It is with immense honor and joy that we welcome you all to the National Conference on Genetics and Genetic Engineering, organized by the Genetic Engineering Association, Department of Genetic Engineering, SRM Institute of Science and Technology, a gathering of brilliant minds dedicated to the ever-evolving field of genetics and genetic engineering. Today, we stand at the crossroads of discovery and innovation where science continues to push boundaries and shape the future of healthcare, biotechnology, and beyond. Before we begin, on behalf of the Department of Genetic Engineering, I would like to extend a warm welcome to all our esteemed guests, the dignitaries on the dais, students, and all the enthusiastic participants who have joined us today. Your presence and participation makes this event truly special. Before we begin to seek guidance, wisdom, and strength in our journey of knowledge and discovery, I now request you all to rise up for the invocation, the Tamil Thai Vaalthu.
நீராறும் கடலொடுத்த நிலமடந்தை கெழிலொழுகும் சீராரும் வதனமென திகழ்பரத கண்டமிது தெக்கணமும் அதிசிறந்த திராவிடன திருநாளும் தக்க சிறு பிறனுதலும் தரித்தனரும் திலகவுமே அத்திலக வாசனை போல் அனைத்துலகும் இன்பமுற எத்திசையும் புகழ் மணக்க இருந்த பெரும் தமிழனங்கே தமிழனங்கே உன் சீரிலமை திறம் வியந்து செயல் மறந்து வாழ்த்ததுமே வாழ்த்ததுமே வாழ்த்ததுமே.
Thank you. In Indian tradition, lighting of the ceremonial lamp symbolizes the dispel of darkness and emergence of knowledge, wisdom, and enlightenment. As we embark on this academic journey, let us illuminate this event with the spirit of inquiry and innovation. I now invite our esteemed dignitaries to come forward for lighting of the lamp. Thank you, sir. Thank you, ma'am. As the lamp is being lit, let this be a reminder that knowledge, like light, has the power to illuminate minds, inspire ideas, and transform lives. With that, we officially inaugurate the NCGGE conference, and now I would like to request Dr. Kiran Kumar, Head, Department of Genetic Engineering, to deliver the welcome address.
Very good morning to everybody. So it's a true pleasure for me to welcome you all to the National Conference on Genetics and Genetic Engineering, 20 years of accomplishments, a conference devoted to the rapidly emerging field of genetics and allied fields. So genetics is the backbone of life. So if you recall the quick history of genetics, so we had era of classical genetics or Mendelian genetics, era of molecular genetics, and era of recombinant DNA technology. Then came the era of genomics. So currently we had era of genome editing, and so genetics has seen a path-breaking progress over the last century, and science of genetics is playing an important role in all walks of modern life. So from gene therapy to personalized medicine to mRNA vaccines in healthcare, genetically modified crops in agriculture, profiling in forensic science, as well as synthetic biology approaches, developing new products like plant-based meat alternatives, etc. So with these exciting developments, genetics remains still a nascent field and lot of innovations, excitement happening. So it's our sincere hope that this national conference will serve as a forum for geneticists to explore groundbreaking advancements in genetic technologies. The conference has 4 domains, namely human genetics and animal biology, genomics and computational biology, plant molecular biology, and microbial genetics. So in each of these domains, we have identified leading geneticists from around the country, and over the next 2 days, a series of exciting lectures has been scheduled. So apart from this moment is a very memorable and precious one for us, the Department of Genetic Engineering, because the Department of Genetic Engineering is celebrating a milestone of 20 years of contributions in teaching and research activities. So our department has a very strong alumni network, and so our more than 70% of our alumni have gone for higher studies, and they are in this domain. They are pursuing genetics and genetics-related research careers. And as part of the celebrations, today we have invited 3 of our distinguished alumni to this conference, and we are going to honor them in this forum. So as the Department of Genetic Engineering is stepping into its 3rd decade, so we are going to focus more on cutting-edge research activities. So we are going to strengthen our alumni network and involve our alumni in teaching as well as collaborative research activities. And another major area which we are going to focus is that we are going to strengthen our industry-academia partnerships by actively engaging with the pharma industries, diagnostic companies, genomic startups, crop science companies, etc. So with that brief background on the conference as well as our department, so I would like to welcome our registrar, Professor S. Ponnusamy, sir, for his presence today in spite of his busy schedule. So he has found time to grace us. Welcome, sir. So I would like to welcome our chief guest, Padma Shri Dr. V. Mohan, for his presence. So he is going to deliver the keynote lecture today. So we are all eagerly looking forward to his talk. So welcome, Dr. Mohan, sir. So I'm also delighted to welcome our guest of honor, Professor S. R. Niranjana, Vice Chairman, Karnataka State Higher Education Council. So welcome, sir. So in spite of his busy schedule, so he has accepted our invitation. So I'd like to welcome our Dean, Professor M. Bharani. So I'd like to welcome all my faculty colleagues, research scholars, participants, external participants from other colleges, and my dear students. I welcome all of you. Let's all have a very good time on the next 2 days deliberating about the advancements in genetics and related fields. Welcome. Thank you.
Thank you, sir. I now request our registrar, Dr. S. Ponnusamy, to felicitate the guest of honor, Professor S. R. Niranjana. Sir, I also request you to felicitate our chief guest, Padma Shri Dr. V. Mohan. Thank you, sir. I now welcome Dr. S. Ponnusamy, Registrar SRM IST, to deliver the presidential address.
Very good morning to all of you. The chief guest of today's function, Padma Shri Dr. Mohan, Chairman, Madras Diabetes Research Foundation. The guest of honor, Professor S.R. Niranjana, Vice Chairman, Karnataka State Higher Education Council. Dr. Parani, Dean School of Bioengineering. Dr. Kiran Kumar, Professor and Head, Department of Genetic Engineering. And my dear colleagues on the dais and off the dais, invited speakers, research scholars, and my dear students, a warm morning once again. Indeed, it gives me a great pleasure to preside over the National Conference on Genetics and Genetic Engineering, 20 years of accomplishment, which is organized by the Department of Genetic Engineering, SRM Institute of Science and Technology. Friends, SRM IST has been constantly progressing in the area of research. We have been supporting the various research activities in different forms. An average of 7 to 8% of the total budget of the university is allotted to research. And in fact, we have introduced teaching track faculty members with reduced teaching workload and an excellent salary package. Their only job is to do research with less teaching workload, and around 120 research assistant professors, associate professors, and professors are working in the university. They bring a lot of laurels in terms of publications, funds, research funds from the various organizations, etc. Their services are laudable. Apart from that, we also provide for the faculty members as well as for the students. And every month, we pay incentives to the research, for research publication to the research scholars. And very recently, we have introduced incentives for the students also, and that is apart from the water we support in the form of stipend. Okay, this total incentives alone comes around 6 crores every year. This is for your kind information. We have been ranked 24th in the country under research category in the NIR of 2024 because of all these contributions. Again, in Shanga ranking, which is also an independent international ranking on the basis of research, our position is 9th to 12th in the country. Also, in Nature Index publication, we are in the 23rd position nationally. And as I mentioned, we invest a lot of money on various activities of research. For example, right now, we have around 200 crores worth of research equipment which are housed at various buildings in the university. And at SRM, we have been supporting the national and international conferences of this type, also workshops and seminars to understand and update the different research areas. Today, we have eminent speakers like Dr. Mohan, and he is well known in the country for his exemplary services. Sir, thank you very much for accepting our invitation, sir, once again. And I request all the students to interact with the eminent speakers and other invited speakers. My dear students and scholars, I hope the discussions and the deliberations that are going to happen in the next 2 days will improve your knowledge in the respective areas. My best wishes for the grand success of this conference. Thank you for this opportunity.
Thank you, sir, for your kind words. I now welcome Dr. M. Parani, Dean, School of Bioengineering, to address the gathering and to introduce the guest of honor, Professor S.R. Niranjana.
Very good morning to all. I am here for my first official function after becoming a dean, and I am fortunate that the first function itself is from genetic engineering, because I am from genetic engineering. I also take this opportunity to sincerely thank all the faculty members of genetic engineering. With all their work, I took the credits for all of that, and I became the dean. So I am very grateful for all the support and the hard work done by the faculty for the past 20 years. So I thank all the faculty. And I also thank the students and the alumni. Today we have some great alumni who published in Nature and all, and that is our dream kind of achievement. It's achievement for ourselves. So that's how we feel. Our family members have published in Nature. And I also thank the management to select a homegrown person for dean position. So with that personal note, I would like to begin my job today to introduce our great guest, Professor S.R. Niranjana. He is a Vice Chairman, Karnataka State Higher Education Council, Government of Karnataka, Bangalore. And Professor Niranjana was born in 1959 in a village in Karnataka, and he has done his BSc at a government science college, and he obtained MSc Botany and MPhil in seed technology and PhD in botany from University of Mysore. I myself work in botany, sir, so we are plant science people. We are very proud to and happy to have you here. He also worked as a postdoctoral fellow at Danish Government Institute of Seed Pathology in Denmark. Professor Niranjana is a former Vice Chancellor of Gulbarga University and former Chairman and Distinguished Professor of UGC BSR Faculty Fellow in the Department of Biotechnology in University of Mysore. He has worked on various aspects of agriculture and sustainable development with his team of research scholars and postdoctoral fellows for 4 decades. He guided 21 PhDs and 40 MPhil students and several postdoctoral fellows. And he published astonishing 220 research articles in highly reputed national and international journals. with more than 7,000 citations. And he also has 4 patents, which includes 2 international patents. And Professor Niranjana is a recipient of several fellowships and awards. So I would like to mention a few. One is the National Academy of Sciences, India, and Fellow of National Academy of Agricultural Sciences, Fellow of National Academy of Biological Sciences, Fellow of Karnataka Science and Technology Academy, Department of Science and Technology, Fellow of Indian Phytopathological Society, Fellow of Indian Society of Mycology and Plant Pathology, and also Fellow of Danish International Development Agency, Denmark. He has also received, these are all the fellowships. Now he has many awards. I'm again going to mention only a few. Sri Visvesvaraya Award and Raja Ramanna Award, Raman State Award, Mehta Memorial Award, National Academy of Agriculture Sciences Recognition Award, Prasad Memorial Award, Binde Memorial Award, and Chalan Lifetime Achievement Award. These are only few. There are many more which I am skipping. He has organized 25 national and international conferences, attended more than 150 national and international seminars, and he has frequently traveled to many countries. He is also instrumental in establishing Asian Seed Health Center by transferring the seed pathology courses from Danish Government Institute, Denmark, to Department of Applied Botany, University of Mysore. He has received more than 200 crores funding. One of single project itself was 100 crore funding. So he has also got a 4.5 crore grant and established a network research and training centers in China Agriculture University, China, Nepal, Vietnam, Bangladesh, Tanzania, and South Africa. So he worked as a various bodies as a chairman, search committee for Vice Chancellor, Bangalore, and also chairman of Research Advisory Committee for CSR, TA, and New Delhi. With this introduction, I once again welcome on behalf of SRM University and myself to this conference.
Thank you, sir. I now kindly request our guest of honor to deliver the guest of honor address.
Good morning, all of you. The faculty and students of the Department of Genetic Engineering, School of Bioengineering, SRM Institute of Science and Technology, is organizing a 2-day national conference on genetics and genetic engineering, 20 years accomplishments. On this occasion, today is the function presided by Professor S. Pannaswamy, Registrar SRM IST, and the chief guest of today's function and keynote speaker, Padma Shri Dr. V. Mohan, Chairman, Madras Diabetic Research Foundation, and my good friend Parani, a dean, and the Professor S. Pannaswamy, the Kiran Kumar, HOD Department, teacher colleagues, researchers, students, media persons, invitees, ladies and gentlemen. I thank the Parani for reading the long journey in the science. Now I am the policy makers for Karnataka state government to make the education policies. I am switch over to the science to the administration, and I served as Vice Chancellor for 4 years. And in 4 years of my time, I produced 5 PhD in biotechnology when I was a Vice Chancellor. So total 21 PhD. And I lost the service of 4 years. During my Vice Chancellor tenure, I would have guided another 5 more students, but I lost that opportunity. Science is great than the administration, what I thought. Administration is a different and science is different. I can see young and vibrant students who are sitting in the group of the galaxy. You have a great responsibility in this country as far as science is concerned. We have started science in this country in 1947 itself, not after the independence. What was the status of science in 1947, 1960s? I will take 10 to 15 minutes, which is going to help you to inspire. That is what the Purani told. I don't want to speak much on science. Today I will speak something is a mix of the science and responsibilities and the status of the science in this country as far as the global contest is concerned. I think Mohan will speak on diabetes. I am also interested because I am the diabetic patient from last 13 years. So that is the part of science. In this country, we have started the education system from the Gurukula system of the last many years. We have started our education. Our historic universities like Nalanda, Taxasila started the quality education with the interdisciplinary and multidisciplinary manner. Today we are talking interdisciplinary and multidisciplinary. It's not a new one. We have started multidisciplinary and interdisciplinary in Nalanda, Taxasila University long back 800 years or 10 years. So those type, the subjects are taught in Nalanda, Taxasila, those so interdisciplinary. Science was not established. Astrology was taught. Politics was taught. Philosophy was taught. With these studies, the Nalanda, Taxasila universities are attracted the many countries for the quality education. Today I am seeing the ambience of SRM. Attracting the students for education is the talent of the institution. Attracting the education from different parts of the country, how you are going to give the quality education is important. That is the way Nalanda, Taxasila has been done. 10,000 students and 2,000 teachers. 10,000 students and 2,000 teachers. See the ratio of teachers. They were taught. So you have gone that education, but because of many reasons, Nalanda, Taxasila was demolished. You know that. I don't want to go in that one. So after the independence, we have taken the independence from British in 1947. Really, the country was faced the problem by that time. You know that there is no communication. There was no food security, and the poverty was in high order in country in 1947 when we have taken the independence from British. After that, the British given the country to us. Jawaharlal Nehru was the first prime minister. Then he thought that these three problems are completely hitting the country. It was very difficult to sustain this country in the beginning up to 1960 when the Swaminathan was brought the Green Revolution. 13 years. Up to 1960. You know that the Dr. M.S. Swaminathan has, how he struggled. Today you have to remember because you are having a genetics department. Jawaharlal Nehru said on that day, see, by experiencing this country must have a science and technology. Only science and technology can eradicate the poverty and hunger of this country. This was the statement made by the Jawaharlal Nehru. What kind of technology with us by that time? We don't have any technology hardly by that time. Committee was constituted and they are decided to start only 4 IITs in this country on zonal wise. Zonal wise, east, west, north and south. 4 IITs was started. I don't want to go in detail. You see that, you try to see you inculcate the practice of updating your knowledge, the history of this country from last 75 years, what we have done. So today we are running 23 IITs in the country, as far as technology is concerned. That technology, the first brought in 1960 by Dr. M.S. Swaminathan is the Green Revolution. That is the technology. Many newspapers are published in 1947. That was said Indians are not having the future because of their growing population. Either somebody has to kill through thermobomb or they have to die going to a slaughterhouse like a sheep. That was the historic transition took place in food security in 1960 by Green Revolution. Today we are reached the status of a gene revolution, from Green Revolution to gene revolution. We have made such a wonderful attempt in agriculture. Today IT is strong and we are sustained in a satellite mission. We are succeeded in space mission. We are succeeded in vaccine. Last 2 years, I think we are all left out by corona. A small virus was completely shake the whole world. And we have developed our own vaccine to sustain ourselves. We must be proud. And we have succeeded that kind of developing a vaccine. We are moving in automatic car today. Green revolution to gene revolution. Drug discovery. Space mission. All these stories are the success stories of the country as far as science is concerned. But the quality research, as far as genetic engineering is concerned, in my experience, I worked on the biological control of a seed-borne pathogens. I am the seed pathologist and a plant pathologist, and then agriculture biotechnologist. Still, a genetically modified crops are in a discussion. We are not able to pass the ordinance or pass the rule for a pipeline crops which are in the GMOs, genetically modified. Why? Many people have told that there is no biosafety in this country. That's what the biosafety is not there for GMOs. If biosafety is not there in this country, then how your pesticides are going to be approved? What kind of parameters you are using to approve the biopesticides? When the biopesticides comes, you are talking on the biosafety issues. Then how you are certifying your chemical pesticides, If biosafety measures are not there, you please send it to somewhere in the globe where the biosafety measures are doing and get it approved from those laboratories where in the globe. So these technologies, we need the people to work on skill basis. That's what the skilled education government is thinking on introducing a skilled courses in undergraduate, and we have taken up many initiatives in skilling of education. Skilled courses has to be introduced to the students for the science, commerce, arts, and languages. What kind of skill? Today, communication is also a skill. You see that the percentage of students are passing in competitive examination is very less. Very less. I don't know Tamil Nadu, Karnataka. I know that the passing percentage is 0.2% of the competitive examination. UPSC examination competitive is 0.5%. Why our students are not able to compete with? Because the skilling and interdisciplinary knowledge in competing with the different subjects to pass the competitive examination, you need the information in your brain in an interdisciplinary manner. You must know the botany, you must know biotechnology, you must know zoology. I'm not telling that you have to read everything in detail. At least I must expose for the subject what is the economics. How economics is going to take part in the science, in technology development, in calculating a cost-benefit ratio. How your technology is going to calculate as the cost. Each and every technology has to be calculated by cost-benefit ratio. How you are calculating your GM technology. That a technology has to be transferred to the field that is calculated by the cost-benefit ratio. How many technologies? That's what a skilling, a technology, economics. Today, each and every subject is important for the social perspective. Economics is important. When you are handling an experiment for DNA isolation, you must know what is the cost involved in one experiment for DNA isolation. Because it's a public money what we are using for the science today. We are all accountable. That's what the technology development and transferring technology is not easy for any scientist to transfer the technology to the field. These technologies are going to be calculated based on the cost benefit ratio and what is your success technologies with you. Today we are looking for. So those technologies are going to help for the society, and after the independence, what kind of technologies we have transferred so far to the society, that is the important challenge for a scientist. Not only for biotechnologists, we are talking in IT sector, we are talking in biophysics, we are talking in a chemical science. This interdisciplinary kind of knowledge is going to help the technology development. That you have to the technology in genetic engineering or in any other science, and how many technologies we are brought out from the a science today. Today, the areas which are concentrating and what is the kind of skills we are using in the a scientific temper. Scientific temper is the big word for this country, which was spelled out by Jawaharlal Nehru long back in 1947. We need the mindset and the temper. Science cannot be managed. Science cannot be managed. Science has to be taken up in a cut-edge kind of a methodology and the technology development. Now it is going to sustain this technology. Everybody is talking today in artificial intelligence. What is this technology? What is this artificial intelligence? How these engineering technologies are going to use as a skill? Now all the engineering technologies are using as a skill. Different skills they are using. That's what the introducing a skilled courses in engineering, introducing a skilled courses in conventional science, introducing a skilled courses in medical science is a need of the hour today for this country. Skilled education is very important for a quality education. And we are talking about the research in this country. I am telling you just one statistics. We are published 1.3 million papers in this country today, as far as 2023 data is concerned. 1.4 million papers of United Kingdom, 4.6 million papers of USA, 4.5 million papers of China. I'm taking only 4 countries. The statistics is telling that now the very important that we have to recall that what kind of the quality and quantity research we are doing in this country. So the number of papers what we have published so far is 1.3 million papers. Those 1.3 million paper is equivalent to UK 1.4 million papers. India research output is the 4th highest in the worldwide. And the H-index ranking USA is 47.02, UK is 39.02, Germany is 36, China is 24.04, India is 14.07. This is statistics. Though we are published the equal number of papers to the UK, 1.4 and 1.3 near to UK number, but UK, the H-index is 39.02, ours is the 14.07. That means more than 70% we are publishing a paper on the quantity basis. We need the papers to make it as a quality paper to increase the H-index of this country. Especially, we are publishing social science, they don't have any this kind of H-index or citations or the publications. They will some number is there for them. But when the scientific journals are ranked, we are spending less money in this country for research. I know that. That is a separate part. I don't want to discuss on this. How much time? See, now the China invested about 4 times more than India. United States invested 75 times more than India on science and technology. So India ranked 46th position in Global Innovation Index in 2021 and 40th position in 2022. So we are spending less money. They are spending more money. See, we cannot compare the Indian universities or any state or public universities comparing to the Oxford, Cambridge, Stanford and other universities. Why our students are not able to get the seat in Oxford, Stanford and Last time I was visited the East London University in UK, whole university was with 90% of Indian students were there. 90%. They went there to join for MBA program, MTech program, MS program. Even some students went for the master program. The students are, I don't mind the student to spread all over the world. Global exposure is one of the skill today. To get the global education, I'm not preventing you to go. The statistics is telling that in 2013, 13 lakh students are left this country. 13 lakh students are left this country. In this year, 18 lakh students are left this country. 18 lakh students went for abroad for education or some other purpose. I'm not telling that moving to the abroad and updating yourself and getting the job and working all over the world, I'm not objecting that. But the intelligence and the ranking in publication, science and technology, we are losing the, I'm not telling you are not good students. They are good students. I'm not telling the 18 lakh students are good students. But we have better students in this country. Whoever is remained in this country, we have a responsibility to run this country, run this show. Whoever is left, we cannot call them back unless we give a food and water for them. We cannot call them. Now government has made, whoever is using the government money to go abroad and for higher studies, they have to come back and serve this nation. They are put the rule now. Whoever went on their own, we cannot call them back. And this kind of education, going abroad, exposing yourself and updating your knowledge. Today, my dear students, not like us, not like the people who are sitting on the dais, our era was different. I don't have the computer by that time when I was completed my MSc program. When I was completed my PhD program, it was a manual typewriter. I was typing my PhD thesis. We are writing an index card on those days by seeing some books and all that, some literature. Today, not like that. Today, you are having a very good facilities and technology, laboratories and scientific equipments to do some good science in this country. Good science in this country. I was attended last time the World Education Forum in London. 140 countries are attended, 140 country higher education ministers and policy makers, and the vice chancellors are attended that forum. Every year it is conducting in London. That World Education Forum discussion was only on the artificial intelligence all over the globe now. How you are going to use this artificial intelligence in science? That's what the biotechnology now is looking for. What kind of science we are looking for in the area of biotechnology now? Innovations, genetically engineered plant to resist environmental stress. Stress is the major part. The ICR is funding money for the stress management from last 20 years. Still we have a problem. We are not managed the stress in plants. Stress management is the one area, challenging area for biotechnologies. And then another one is a cell-free protein production platform. Cell-free protein production platform. Then the reproductive hormone from cows for cows. New strain for E. coli for the synthesis of superior PHA. I don't know how many people are aware of all these terminologies and the research. That's what I told. Research is the backbone for the science, and research is going to become the education of the science. Then broad range of biodegradable biosurfactants, new methods for controlling gene expression in agricultural biotechnology. Everybody is studying on the gene expression. That gene expression you have to study at the point of agriculture. Agriculture sustainability is very important. I was telling that in 1960, when Swaminathan was brought the Green Revolution, we have produced 50 million ton grains on that day. Today we are producing 350 million tons. We need 203 by 2030, 400 million tons for our growing population. You must know the statistics. How much required food for this country? So that we have produced a good number of grains in this country. Food is no problem another 10 years, 20 years, my dear students. So don't worry about the food. You will get food in this country. Food has been sustained and we have developed very good varieties and we are producing a good quantity of seeds for a food. that our challenge is the education and the science and the technology. Our challenge part is education, science, and technology. That's what genetically, this is another part what we are recycling plastic with synthetic organisms. Then genetically modified fiber crops to make a waterproof materials. Universal plant gene modification for more efficient growth. Then naturally occurring biocompatible proteins for a tenable proton conduction. Then a current hot topics in biotechnology is artificial intelligence. How you are going to use this technology? You see that many people are telling that when artificial intelligence comes, man is not having any job, everything will do by robotics. I think Mohan can address all these things, how robotics are going to help us now. Then the genome surgery, that is the gene editing. Then gene sequencing, personalized medicine or synthetic biology, biomanufacturing, bioprinting, microfluids. All these areas are coming up in the genetic engineering now, genetics, that you take this as a challenge part in these areas. And this genetic engineering has opened up opportunities for increasing crop yield. That's what our agriculture production is increasing now. We are sustained in producing agriculture productions now. IT and agriculture, we are in the top in this country. IT and agriculture is the top. So now we have to use these applications in improving a crop yield in agricultural technology and in the reducing crop losses to insect disease and post-harvest storage problems and enhancing the nutritional value of some crops. Today, your nutraceuticals is one of the area. See, everything you are taking together, you cannot achieve anything, my dear students and colleagues. See that what is SRM University, for example, you are having a genetic engineering department. What is your thrust area, what you are targeting? One should identify and recognize your university on one area of genetic engineering, where you are the masters. Don't take it everything in hand now. You are not going to succeed in that type. We have a very vast area of science today as far as biotechnology is concerned. In 92, we have started biotechnology in Mysore University. We are the first of started MSc biotechnology. We are very first in this country. Last 30 years, then we got the DBT recognition for biotechnology, that DBT HRD program. It was given very small amount for the technology. Today, money is important to make the science. Today, money is important to make the science is expensive technologies, expensive instrumentations. One NMR, the cost is 6 to 7, 8 crores. Everybody cannot afford, every student cannot expose for the equipment. X-ray diffraction is close together. LCMS is close together. So my idea is, we are making the policy now. Wherever the equipments are there and it is accessible to all kind of students to go and use these equipments for their knowledge purpose. This is called as sharing of facilities, sharing of knowledge and disseminating the knowledge for others. So that is the very important area in biotechnology, and take this as very important a subject today as far as technology is concerned. Keep all these things in your mind in the modern new biotechnology, what is the importance of this genetic engineering. And the environmental biotechnology is also there. That where they are concentrating on the bioenergy. All these areas are very important area to contribute to the science. And the problems are there. Really this biotechnology is going to solve the societal problems. Really this biotechnology is going to solve the health problems. Really it is going to solve our crop production problems. And you have to address properly by using the proper technology for science and technology development. And this is the status now. Whatever we have succeeded, but that success also we need the skilled workers, skilled students to work for this technology, to sustain this technology in this country. That is a big problem for us now. The student after doing his PhD, he don't want to touch any equipment for a skill. So everybody wanted to become a scientist. I know, see, in Germany, you go in any European country or any American country, professors themselves, they are preparing the media, professors themselves, they are working, and everybody is working together. And many students are work like a technicians. Apart from my knowledge, apart from my skill, I can work. But don't try to manage together. You try to update your knowledge independently as far as science is concerned. You cannot. I've seen the students preparing the media, 8 students together and preparing media. One fellow is bringing water, one fellow is weighing chemical, one fellow is not like that. You have to make yourself update the complete knowledge of the experiment. Then only the science can be moved together and move further. We cannot share that kind of things. And these are all the important challenges. How this we have started science and technology. What is the importance of science and technology in India? Now the technology is concerned, where the genetic engineering is taking role in that one. And these biosafety issues are how you are tackling the biotechnology, techniques now. All these you keep it in your mind and come up in your life. You have a better life than any science. And today, without, my dear students, without chemistry, there is no biology. Without chemistry, there is no biology. You try to inculcate to understand the chemistry of the science. Your bioactive molecule, your mechanism. Biochemistry is taking a very major part in technology development. So that's what I'm telling, interdisciplinary knowledge is required today for the technology development. Inculcate all these practices. And all these things are going to become the policies now. No technology can be passed very easily unless you define. I know that the patents, I read the Parani has read, I have 4 patents. I have took the 6 years to pass that patents. My files are this much to make patents on biological agents to control insect pests. So technology development through patent is a very horrible attempt. So you have to reach that kind of science, then only it is help this type of technology to this nation for future. And you are all future scientists, my dear students. You have a big responsibility. I hope SRM University will provide the facilities and the ambience, very good ambience. I have seen first time I came in the university. Many of my colleagues were telling, you are going to a very good place and see that it's a very good university. After coming over morning, I saw that very good ambience. I have not seen any public universities like this even. Very good university. We have a setup. Private partnership, private and public partnership is going now like this only. Now everybody is talking, foreign universities are coming and establishing in India, and UGC has permitted them to come and establish foreign universities. I don't mind. This kind of policy issues are going in this world. Going abroad, the students, and coming inside the country. Many people wanted to learn here, and many of our students are going out to learn there. These are all the, in that, whatever the facilities, whatever the, in a stipulated time, you have a 2 years of master program and 4 years of PhD program. In this program, you try to inculcate the practice of learning and updating of your information and the knowledge. It is going to help in your career. So with these words, I thank Parani for inviting me and sharing some of my views and thoughts. I've not thought much on science, but the status of science in this country and the duties of your as a scientist in this country. And we have challenges as far as technology is concerned, and we have some requirements for this country as far as science is concerned that I have explained, and try to inculcate all these things and keep these things in your mind and inspire yourself and do some good work. Then knowledge is going to help for the technology development in this country. I thank once again the registrar and the Parani and other colleagues who are in the department and inviting me and sharing my thoughts in this a fine morning. Thank you very much.
Thank you so much for your kind and inspiring words, sir. I now request our guest of honor and chief guest to release the conference proceedings. Thank you, sir. As we are celebrating 20 years of accomplishments of the Department of Genetic Engineering, we are honoring our distinguished alumni with awards such as Young Scientist Award and Young Entrepreneur Award. I now call upon Dr. Rex Arun Raj to announce the awards.
The legacy of a great department is not in the books, but in the success of their alumni. I am privileged to preside over the award ceremony. Distinguished guests on the dais and off the dais, today we have amongst us three of our alumni, Dr. Anant Jain, Assistant Professor, Center for Higher Impact Neuroscience and Translational Applications, Dr. Devi Vaishya, Assistant Professor, ISER Bhopal, and Mr. Sujit Kumar Singh, Founder and Director, Cytogene Research and Development Technologies, Lucknow. In the past year, the department has been reverberating the success and achievements of our alumni, celebrating them with distinguished awards. Department of Genetic Engineering is like a flourishing garden where the seeds of knowledge, values, and dreams are sown carefully. Now, the seeds have blossomed into towering trees of success, their branches stretching far and wide, a season to bear fruits and wisdom of knowledge, judgment, and achievement. The teachers who nurtured them, now they stand proud as gardeners, witnessing the harvest of their labor. Each alumnus is a testament to the commitment of the faculty, quality of education provided by the department, and the vision of the management. Today, they return not as students, but as the voice of SRM, ready to sow seeds for the generations to come. On behalf of the Department of Genetic Engineering, I now invite Dr. Anant Jain onto the stage to receive Young Researchers Award. I request Professor S.R. Niranjana, Vice Chairman, Karnataka State Higher Education Council, Bangalore, to do the honors. I request the distinguished members on the dais to join him. Now, I invite Dr. Devi Vaishya, Assistant Professor, onto the stage to receive the Young Researcher Award. I request Padma Shri Dr. V. Mohan to do the honors. I request the members on the dais to join him. Not only we produce researchers, we also produce entrepreneurs. Now I invite Mr. Sujit Kumar Singh onto the stage to receive the Young Entrepreneur Award from the Registrar, Professor S. Ponnusamy. I also invite all the distinguished members on the dais to join him. Ladies and gentlemen, please give them a big round of applause. This concludes the award ceremony. Thank you.
Thank you so much, sir. Next up, we have an exciting industry partnership signing of the MOU. I call on to the stage Dr. Arul Jyoti to brief us about the industrial collaboration.
Good morning, everyone. This is an exciting event. It's a proud moment for SRM Institute of Science and Technology as we take another step forward in bridging academia and industry. Today, we formalize a partnership that will enhance the academic learning and practical training of our students in the field of genetics and genomics and computational biology. Through this collaboration with Anderson Diagnostics Private Limited, our students will gain access to industry-driven expertise, hands-on training, and research opportunities in cutting-edge genetic testing and counseling. Now, it's my honor to invite our esteemed guest from Anderson Diagnostics to the stage. First, I invite Dr. Surya Kumar, Director and Radiologist, Anderson Diagnostics, a visionary leader in diagnostic services, to join us on the stage. I'd like Dr. S. Purnaswamy to felicitate our guest. Next, I welcome Dr. Fiona D'Souza, Chief of Scientific Operations, whose expertise and leadership drive innovation in genetic testing and genomics. I would like Dr. V. Mohan to present a bouquet to Fiona. Thank you, sir. I also would like to invite Mr. Rajendra Kumar, Global Marketing Manager, whose strategic initiatives have played a key role in strengthening our partnership. I'd like Dr. Parni to felicitate Mr. Rajendra Kumar. Next, I welcome Ms. Shruti Abhirami, who is an alumni and also as a genetic counselor working in Anderson Diagnostics, to come out on the stage. I'd like Professor Kiran Kumar to felicitate Shruti. Let's all give them a warm round of applause. I'd like to invite Sri to highlight the MOU.
The MOU between SRM IST and Anderson Diagnostics aims to enhance academic industry collaboration by providing hands-on training, internships, and industry exposure for students in genetics and genomics. It also facilitates joint research, guest lectures, and a value-added course on genetic counseling. Through this partnership, Anderson Diagnostics gains access to a talented pool of students for internships, training, and potential recruitment. Additionally, they benefit from collaborative research projects, access to SRMIC's academic resources, and knowledge exchange with faculty and researchers. This marks a significant milestone for the Department of Genetic Engineering as it celebrates its 20th year. Let us wish this partnership continued success in achieving greater heights. I request Honorary Registrar SRMIST and respected Director of Anderson Diagnostics to exchange the MOU. We extend our heartfelt gratitude to our Honorable Registrar SRM IST for his support in making this collaboration possible. A special thanks to Dr. Surya Kumar of Anderson Diagnostics for this valuable partnership. We sincerely appreciate Dr. Ramya, Dr. Kiran Kumar, Dr. Arul Jyoti, the Law Officer of SRM IST for their dedication, and Dr. Fiona D'Souza, Mr. Rajendra Kumar, and Ms. Shruti Abhirami of Anderson for their seamless efforts in bringing this MOU to fruition. Thank you for all your invaluable contributions.
Thank you, sir. I now call upon Dr. Devi, Organizing Secretary, NCGG, to give us the inaugural vote of thanks.
Respected dignitaries, esteemed guests, faculty members, and dear students, I consider it a privilege to extend the vote of thanks on this momentous occasion on the inaugural function of the National Conference of Genetic and Genetic Engineering, celebrating 20 years of accomplishments. I extend my deepest gratitude to our guest of honor, Professor S.R. Niranjana, Vice Chairman, Karnataka State Higher Education Council, for accepting our invitation and gracing this occasion. A heartfelt thanks to our Chief Guest, Padma Shri Dr. V. Mohan, Chairman, Madras Diabetic Research Foundation, Chennai, for accepting our invitation and consenting to deliver the keynote address. I also thank our sincere appreciation to Professor S. Ponnusamy, Registrar, SRM Institute of Science and Technology, for presiding over this inaugural function and offering his invaluable guidance and support. I also thank our Dean, Professor M. Parani, for his unwavering support and encouragement, our HOD, Dr. Kiran Kumar, for giving us a warm welcome, and to all the colleagues and my students for extending your support for this conference. I also have a special note of gratitude to our esteemed sponsors, Anusandhan, the National Research Foundation, Government of India, and the Department of Biotechnology, and our industry sponsors for the generous support and encouragement in organizing this conference. I also like to acknowledge Anderson Diagnostic Services Private Limited for the industry partnership through the MOU. Now I am cutting it short. So thank you all, the external participants, for being here, and we hope that you'll have an engaging, rewarding experience for the next 2 days. I now request all the dignitaries on the dais to occupy the front seats for the sessions to begin. Thank you all, and we are waiting to hear Dr. Padma Shri, Dr. V. Mohan's lecture. So thank you all.
Thank you to all the dignitaries. Once again, I welcome you all. Let this event be a celebration of knowledge, innovation, and collaboration. Without any further ado, let's begin the first session, the keynote lecture. I now call upon Dr. Satish to introduce to us our chief guest, keynote speaker, Padma Shri Dr. V. Mohan.
A very good morning. Wow, what a start we have. So I am delighted to introduce Padma Shri Dr. V. Mohan. First of all, we are very grateful to Sir for kindly accepting our invitation to be the chief guest of this conference and to deliver the keynote lecture, despite the hectic clinical schedule. He's a distinguished diabetologist, an eminent scientist, and a humble human being. So he has over 5 decades of experience in diabetes care, research, and education. He serves as a chairman and chief of diabetology at Dr. Mohan's Diabetes Specialty Center in Chennai, which is recognized as the Center of Excellence in Diabetes Care by the International Diabetes Federation. So his insights and expertise will undoubtedly inspire us to continue our mission towards better diabetes care and overall health. So now I invite him to deliver the keynote lecture entitled Genomics of Different Forms of Diabetes, Clinical Relevance to it. Please, sir. Sir, thank you.
Respected guest of honor, Professor S.R. Niranjana, Professor Ponnusamy, Registrar, Dr. Kiran Kumar, Dr. M. Parani, Dr. Devi, respected faculty, students, ladies and gentlemen. Thank you very much for the invitation to be here today to deliver the keynote address. I'm a clinician, but I also do a lot of research. I would like to thank Dr. Kiran Kumar and Dr. Ilangovan for inviting me, and congratulate all of you for completing 20 years of stellar work in genetics and genetic engineering. I'm sure the best is yet to come, and I wish you all the very best going forward.
the guest of honor has told you that it's not just the quantity of the publications and the research that you do, but the quality, which is also equally important. So I know we are slightly behind time, so I'll try to give you a bird's eye view. I'm a clinician, so I'm going to talk about diabetes, the area in which I work. And diabetes itself is such a big group of disorders. It's not one disease, it's several diseases. So I'll try to give you an overview of the genomics of different forms of diabetes. But in the second half of my talk, I'll talk about the genomics of monogenic forms of diabetes, which has a single gene defect. And that is where in the clinic itself, we use genomics even for treatment. I have no conflict of interest. These are all government-funded studies. I'd like to start off by thanking my head of molecular genetics, Dr. Radha Venkateshan, and her team. This one department itself in our institute in MDRF has produced 150 papers just on the genomics of diabetes itself, and 16 PhDs have completed from this one department. Totally 75 PhD students have already got their PhD from our institution, and our total number of publications now stands at close to 1,800 publications, 1,800 publications, and my own personal H index is 157. So I thought I'll just tell you that just to inspire the students here. So the flow of my talk is as follows. I'll talk a little bit about the genomics of type 1 diabetes and type 2 diabetes, the commonest form of diabetes, gestational diabetes, fibrocalcific pancreatic diabetes. I won't take time. I'll just show you one or two slides of each of this. I want to talk a little bit more about monogenic forms of diabetes, because that is where it's truly life-changing and game-changing, where genomics actually completely changes the treatment. So let's start with type 1 diabetes. Type 1 diabetes, you know, usually occurs in children. Can occur at any age, usually above 6 months of age. Below 6 months, we have neonatal diabetes. We'll talk about that. But this usually occurs in children, but can occur at any age. It can even occur at older people, 50, 60, 70 years, but it's very rare in adults. In children, it's quite common. People with type 1 diabetes have zero insulin in their body, not a drop of insulin. And therefore, they have to take insulin injections 4 to 5 times a day if they have to be alive. If they stop the insulin, they'll die. In fact, insulin was discovered only 104 years ago. And up to 104 years ago, a child with type 1 diabetes lived only for 2 weeks or 3 weeks. 2 months was a long time. 1 year was a miracle. Nobody lived for 1 year. If a child developed type 1 diabetes, a death sentence, worse than the worst cancers that we have today. From there, after the discovery of insulin, children started living longer and longer. And today, I have people with type 1 diabetes whom I'm treating who have crossed 70 years of diabetes, and they're still alive. Some of them have crossed 90 years of age, and they're still doing well. But if you think about the genetics of type 1 diabetes, the primary genetics is related to the HLA system, histocompatible leukocyte antigen system. But today we have genetic risk scores, and these risk scores, the TD type 1 GRS or type 1 genetic risk score, was developed at the University of Exeter by Richard Oram and others. But we now have some studies from India on that. Initially, it started with a panel of 9 SNPs, then 30 SNPs, and now 67 SNPs is what we use. We have exactly adopted because this is the best in the world. So we've adopted that whole thing, and we're now doing it at MDRF, and our initial studies are just getting published. We've been using the 67 panel. Actually, you don't need genetics to actually diagnose type 1 diabetes. A child develops ketoacidosis and coma, it's 99.9999% it's type 1 diabetes. Then you've got a simple test called C-peptide. You do that and it's absent, the diagnosis is very clear. On top of that, you've got antibodies, the GAD antibodies, zinc transporter antibodies, and IAT antibodies. They'll be positive in about 70 to 80% of type 1s. Over and above that, if you still want to diagnose, you can use the type 1 genetic risk scores. It's a pure research procedure. We don't need this in clinical practice. Type 2 diabetes, we all know, has both a genetic component as well as an environmental component. In fact, type 1 and type 2 diabetes are called as polygenic diseases. Why polygenic? Because there are many genes which are involved. They are not causal, they're only an association. You can say this gene is associated with this, but it's not causing it. In monogenic diabetes, actually it's a cause. If you have a genetic defect, that is a cause of the disease. So that's how it differs. The other thing about type 2 diabetes, if you take 100 people with diabetes, 90 to 95 will have type 2 diabetes, and all the other types put together is only about 5% or 10%. Now, in type 2 diabetes, the other thing is that genomics plays a role, but it's only about 40%. 60% is due to environmental factors. It's due to obesity. It's due to lack of exercise. It is due to lack of sleep. It is due to overeating. It's due to too much of carbohydrate being taken. Many factors, physical inactivity, all these produce diabetes on a background of genetic predisposition. If father and mother has it, you will get it earlier. If your father and mother don't have it, you'll probably get it much later, maybe after 50 or 60 or 70 years. Both your parents have diabetes. In India, if father and mother are both diabetic, by age 50, 95% of them will develop, of the children will develop diabetes. Only 5% actually escape. But then still driven by various environmental factors. Today we have about 700 genetic variants which are associated with type 2 diabetes. So it's not necessary to measure all these 700 varieties. Of course, some of them I'll show you, some of them are associated with insulin resistance, some of them are associated with insulin secretory defect, and the list is growing. But none of these actually cause the diabetes, they're only associations. This is one of our early publications where we looked at the TCF7L2 gene. This is the strongest gene associated with type 2 diabetes, and able to show that in those with type 2 diabetes, it is much common. These are other studies on the TCF7L2 gene. If you're looking for one gene which is associated with type 2 diabetes, it will be the TCF7L2. That will be the top one. And then, of course, there are many, many others. As I said, there are 700. This is another gene called as the adiponectin gene. Adiponectin is a friendly hormone. You know that if you have more adiponectin, you won't have obesity, you won't have diabetes. If you have less of adiponectin, then you get it. If you have a genetic defect in adiponectin, then you don't produce enough adiponectin. That's one of the causes of diabetes. Very important gene is called as the FTO gene, the fat mass gene. Now, this fat mass and obesity gene, very interestingly, the FTO gene is associated more with obesity in the European population and more with diabetes in the Indian population. So my colleague, Dr. Ranjan Yajnik from Pune, and our own studies have shown this. Now, this is just a summary of all the genes, some of the genes associated with type 2 diabetes. And you can see that the obesity gene, there's only one major gene, that's FTO gene. With insulin resistance, there are about 3 genes. But look at the number of genes which are associated with beta cell dysfunction or beta cell mass. So the majority of the genes which are associated with type 2 diabetes are related to insulin secretion. So if you have a defect in those genes, you don't produce enough insulin. And that's the reason why you get it. Whereas insulin resistance and obesity are driven more by environmental factors. Eat more, you don't exercise, you put on weight. So those are not really genetically determined. Whereas beta cell function is genetically determined. Now, genome-wide association studies will help to throw up new genes. See, the other genes, when you're going FTO gene or TCF7L2, you take one of those genes, they are hypothesis driven, which means you say, okay, this must be associated with insulin resistance, therefore this gene will be there. That's a hypothesis driven. If you are looking at it not from a hypothesis driven, but a hypothesis generating, then you have to do either a GWAS, a genome-wide association study, or a whole genome sequencing. Then you will pick up new genes which have never been described. 3 GWAS association studies have been done in Indians, and we have been associated with all the 3 GWAS studies. For example, in this one which we published in Nature Genetics, we picked up 6 novel genes. These 6 novel genes were associated with type 2 diabetes only in Indians, because in this cohort of some 70,000 people who underwent the GWAS, we had Chinese, we had Japanese, we had white Europeans, we had Indians, we had everyone. But only in the South Asians, Indians, Pakistanis, Bangladeshis, only they had these genes. We'll say, oh, then you solved the problem. We haven't solved the problem because these are very small effects. The Lord scores are so small that they are not actually the cause. The cause is something else, but they are very strong associations. So here is yet another one. This was done completely no, this is done in Punjabi Sikhs, and then we had one more from Delhi with IGIB. We did this 3. So we have 3 Jiva studies. Each one of them, this one picked up one gene, the TMEM gene. So small incremental knowledge is done. It's very costly to do a genome-wide association study on 50,000, 80,000 people. It's very expensive. At the end of it, you'll pick up one gene. And that gene doesn't tell you much about what the cause is. So in type 2 diabetes, again, it's not necessary to do genetic studies because 60% of it comes from the environment. Even a simple family history. Based on our work, 20 years of our research, we finally developed a simple risk score. It's called as the MDRF Diabetes Risk Score, Indian Diabetes Risk Score. Some of you might have seen this. It just needs 3 questions. Does your father and mother have diabetes? What's your age? And do you do physical activity or not? 3 questions. It costs 0 rupees. And then all you need is a waist measurement, which again costs 0 rupees. You can do the whole thing in 2 minutes. We found that if you do the IDRS, these 3 questions and the waist measurement, it actually beats a TCF7L2 gene. So you don't need to do genetic testing at all. With 0 rupees, you can actually identify. That's not the case with monogenic diabetes. We'll come to that. So here is another one more recently where we looked at young onset diabetes and beta cell function. This is a more recent one, collaboration with the University of the Queen Mary University in London. Very interestingly, we found that why we have this beta cell function, why are Indians more prone to diabetes? We have the highest susceptibility to type 2 diabetes, and yet we are quite thin. And yet we have a lot of diabetes, much more than the white Europeans. Why is that? So we started looking at the genetic predisposition to insulin deficiency, and we found that the South Asians living in the UK as well as the Indians living here, our insulin deficiency genes seem to be affected more than the white European population. Very clearly, we are able to separate out the Indians and the Europeans. You can see it here. This is the Indians and this is the Europeans. As far as the beta cell dysfunction genes are concerned, they're quite nicely separating out. And this is yet another recent publication where we looked at why do we get it at young age. We seem to get type 2 diabetes about 20 years earlier than the white man gets it. White man gets it at 50, 60, we get it at 25, 30. Why is that? We are able to find that certain genes, TCF7L2 being one of them, CDCal being another one, these are all explaining why we are getting it at a younger age. Gestational diabetes is a type of diabetes which occurs during pregnancy. So the lady before pregnancy may not have diabetes, and after she gets pregnant, suddenly they develop diabetes. It can occur in early pregnancy when it's called early GDM or early gestational diabetes. It can occur in late part of pregnancy. It's called late GDM. So 2 types of GDM there are, early and late. We know that there are some genetic factors which are associated with GDM, a number of these genes, including TCF7L2, but we have found out other genes which are associated with, this is one of our publications showing some new genetic variants associated with GDM, and this is yet another one, the CDCal and the HDAC gene. These are all associated. This is a hexokinase domain containing, or the HDAC gene. This is strongly associated with the GDM, much more than even the TCF7L2. So some discoveries in this we have made. Now let me go on to a condition on which I worked many, many years. In fact, my PhD and my doctor of science, everything was on this very rare condition. It's called fibrocalculous pancreatic diabetes, found only in India and a few tropical countries. India has the highest prevalence of the whole world. You develop stones inside the pancreas. So where the insulin is supposed to be secreted, you get stones, just like kidney stones or gallstones, you get pancreatic stones. Those stones produce inflammation, they produce diabetes, they produce digestive problems, and it's called fibrocalculous. Fibrosis means shrinkage, calculus means stones, so fibrocalculous pancreatic diabetes. So we find that this FCPD has some genetic associations, and we showed that one particular gene, the SPINK gene, is the reason why these people get this fibrocalculous. It's a very rare type of disease, and after spending 18 years and publishing some 200 papers on it, I gave it up. I said, this is too rare, we concentrate on something, and the disease is going down, because with better nutrition, the disease actually started going down. These are some of the publications on the SPINK gene. Now, let me move to the more exciting area of monogenic diabetes. So as the name itself suggests, a single gene only is involved. And all you have to find is that gene. And once you find the gene, the treatment changes, completely changes. So this has to be incorporated in the clinic itself. And we have to use it as part of this, not for research at all. This is meant for treatment of patients. Now, there are different types of monogenic diabetes. One of the common ones is called maturity onset diabetes of the young, or MODY. This was described by a man called Professor Stephen Fayance at the University of Michigan. He used to be my childhood hero. And at the age of 93, he was still publishing papers. I knew him very well. And I have the honor of delivering the Stephen Fayance lecture in June this year at the University of Michigan. This term was actually described by him, the maturity onset diabetes of the young. There's another one where the mother and the child will have diabetes and they'll have deafness. So it's called maternally inherited diabetes and deafness. From the deafness, you can pick up that it's this type of diabetes. Then there are many genetic syndromes. I won't talk about that. I will talk about neonatal diabetes, because this is a type of diabetes which occurs just after birth. Even type 1 diabetes comes only by 1 year of age, or this occurs immediately after birth. Within 2 days, 3 days, 1 week, the child gets diabetes. Very, very exciting type. And the opposite of that is the low sugar which occurs after birth. It's called congenital hypoglycemia of infancy. I'll talk about these two and about MODY. So as I said, MODY was described by Tattersall and Fayance, Stephen Fayance at University of Michigan. And this was at that time when they thought, before they described this, any young person who got diabetes given insulin, they thought it's type 1 diabetes. Until Fayance said, hey, hang on, there are some children who don't need insulin. And then he described certain conditions. And I started work on this many years ago. Then when the genetic discoveries came, 14 different types of MODY were found out. It's not one type of MODY, but 14 types, of which this MODY 1 and MODY 3 are the most common, and they are the ones you should look for, because they are the ones which will respond to tablets. They're wrongly treated with insulin. They think that they're type 1 diabetes. If you find out this MODY 1 or MODY 3, they can stop the insulin and be converted to tablets. MODY 2 is even better. You can stop all treatment, because MODY 2 will never develop complications. So once you diagnose MODY 2, you can stop all treatment. You can tell them to go home. They don't need any treatment, okay? Some of the other types are rarer. We won't talk about that. So we have been doing a series of publications on MODY 3, on MODY 1, on MODY 2, on MODY 5. We've discovered a new type of MODY from South India. I won't talk about that. And this is another review where we looked at MODY. You may say, why bother about all these rare type of diabetes? I'll show you why you should bother about that. As a clinician, why I bother about that. Let me give you a story. There's a 16-year-old girl who was diagnosed to have diabetes, okay? She was thin and she presented with severe diabetes. The moment you're 16 years old and you're thin, you're immediately diagnosed as type 1. And you're told that you have to take 4 times insulin, rest of your life you have to take 4 times insulin. Somehow this parents came to us, HbA1c is also very high. Parents came to us and said, can you do anything for our child? So of course, sometimes it turns out to be type 1, then we can't do anything, we just educate them and then send them back. But this particular girl, when she came, the first thing we do is we take a family history. This doesn't cost any money. Just need a paper, pencil, and you can draw it. This is the girl who came to us. She was 16 years old. What we found was that her mother also had diabetes, which is interesting because type 1s, generally the parents don't have diabetes. So that rang a bell. So then when we looked at it further, we found that the mother's sister, all these people had diabetes. If you look at the mother's father and mother, they had diabetes. When you look at their father, they also had diabetes. So one generation, second generation, third generation, fourth generation, there's a fourth or the fifth generation. This is what Stephen Fayance described. He said it will come in families and it will come generation after generation, and they may not need insulin at all. They can be controlled with tablets. So having known this family history, which is very, very strong, very unusual for type 1 to have it. You can see the father side, there's no diabetes at all. Those who are having the red there are in-laws. They are not the father's brother and sister. But the mother's side, it has come like that, okay? So then we did the genetic testing. First of all, we did the C-peptide assay. C-peptide will tell you whether there's insulin secretion or not. In type 1, it will be 0. Here, she had insulin secretion. So again, we said, hey, this is not looking like type 1 at all. Then we did the GAD antibodies and zinc transporter. And type 1 it will be positive. Here it is negative. So we said, okay, we're getting more and more evidence. So then, because of 3-generation transmission, we Center for Genetic Testing. We found this particular gene, the HNF1 alpha, which is MODY3. This is the commonest form of MODY, and this is the one which responds to tablets. It completely responds to sulfonylurea. The cheapest tablets which you have today in the world is the sulfonylureas. And they are the ones to which they respond very well. Insulin is expensive, and you have to inject it several times a day. So we found this mutation. So then, by the time, those days, it took some time. You can see it's 2009 publication in JCEM. By the time we complete our technology was slow at that time. So by the time we got the results, her sister also developed diabetes. Okay. So we said, okay, now we'll test all of them. The star means that they have MODY. Okay. So she has MODY. She also has MODY. It's, by the way, MODY, not MODI. Okay. So the mother had MODY. Everybody who was in red in the mother's family had MODY, okay, including one of her sister's children. This boy here, her cousin, he also had. Here, of course, they were dead. We couldn't get. So everybody who did not have diabetes did not have the mutation. Everybody who had the diabetes had the mutation. So it's like a detective puzzle now, okay, like Sherlock Holmes. You'll say, yeah, so she has MODY. She has developed it from the mother, okay, and you can also say that the ones who have got it here now, their children will also develop in the future. So you can also do genetic counseling. You can start looking for those children early, and you can start treating them early and diagnosing them early. So much can be done just by a simple genetic test. Okay. So what did we do? We took her off insulin. We started her on sulfonylurea. Her sister also responded to sulfonylurea. This, I think, happened in 1980s when I first saw this girl. 40 years later, she's not had a single shot of insulin. She's still treated with tablets. And this is the power of genetic counseling and genetic testing, okay? And this is what we call as precision diabetes. I want you to clap louder a little later on when I show you a more exciting story, okay? Right. So now only some form of MODY respond to sulfonylurea. MODY1, MODY3, and MODY12 respond. So you should pick out these MODYs only. So how do you pick them out when a person comes to you? How do you know, unless you do genetic testing? Genetic testing is expensive. So who to send for genetic testing? The world had not told us that. So we have to now find out who to test. So one of my PhD students, Aarti, I put her on the job. And what we did was find out the actionable MODY, those who can be respond. If you're not going to respond to tablets, why even do the genetic testing? Just continue the insulin, okay? So we found this out. We published this in Nature Reports, where we looked at various tests. So we took MODYs, we took type 1s, we took type 2s. You can see MODYs are not so common. Type 1, type 2 is more common. And then we looked at various parameters. We looked at age, BMI, family history, HDL, HbA1c, and we found that the C-peptide actually seemed to be the best to distinguish, because the blue here is type 1, the green is type 2, and in the middle is MODY, okay? What do you mean by stimulated? They give a fasting blood, then they take breakfast, and at the end of 90 minutes, 1.5 hours, we take one more blood. This test was devised by us 30 years ago, and we still use this. So by just doing this test, this comes out very good. If you look at the sensitivity and specificity of the simple C-peptide, we get it in like 1 hour. If you give the blood in the morning before the patient walks into my room, we already have the results. So the sensitivity is, specificity is 100%, and the sensitivity is 97%. So by just doing the simple test, which just costs 200, 300 rupees, you can say whether it's type 1 or type 2. So if anybody comes to me and say, tell me whether I'm type 1 or whether to take these 5 injections, just with this one test, we can say whether there's possibility of being MODY, whether there's possibility of being type 2, or whether it's type 1, okay? Now I come to the most exciting part, and hold your applause until it's over. Okay, so neonatal diabetes is far more exciting than a 16-year-old person coming. This is a newborn baby, okay? And this baby is just born and is told that they have diabetes. 99.99% all over the world, they start them on insulin, because everybody knows about type 1. They don't know about neonatal diabetes. One point I want to tell you, and I want you to spread this message, is that type 1 almost never occurs before 6 months of age. Almost never. I've seen like one case in my whole life. So you can say it never occurs, okay? It will be neonatal diabetes below 6 months of age. 50% of all those babies who are below 6 months of age will have mutation, which if you detect, you can change them over to tablets. At 6 months of age, they can stop insulin and powder some tablet and give it to them. They'll be done, okay? So here is another story. This is a 3-month-old female baby. She came from Calcutta. She was diagnosed there. So the physicians in Calcutta had heard of us and they said, we'll send the blood sample to Chennai, where Dr. Mohan and colleagues will look at it and tell you whether it's neonatal diabetes. So she came, she was already started on insulin. She had taken 14 units of insulin. For a newborn baby, this is very high dose, but her sugars were not coming down, 300, 400 all the time. So she came to our center. By the time we could do the test, the parents came off with the child and said, we couldn't wait, so we have come here straight. Any other test you want to do, you do for this child, okay? So, okay. So we ordered the genetic testing, and lo and behold, she had this KCNJ. So there are 2 mutations which will respond to sulfonylurea. KCNJ11 and ABCC8. ABCC8 is a sulfonylurea receptor. KCNJ is a potassium channel receptor. So if one of these 2 mutations is there, tablet will work. So every pediatrician who refers to me, including Dr. Raghupati, was formerly in CMC Vellore and now in Bangalore, he says, I pray before I send the blood sample to you. I pray that they should have one of these mutations, and then I send the sample to you, okay? Because they don't have the mutation, 50%, then they have no use. They won't respond. Only half will have the mutation. So this child had this mutation, so we started her on tablet. This is the cheapest tablet in the world, as I said, lebenthlamide. The dose went up. Let's see what happened to her sugar. So this is the dose of the sulfonylurea, slowly increased. This is the dose of the insulin, okay? So dose of insulin from there, that's the insulin dose. Insulin dose come down, come down. By 4th day in the hospital, we stopped the insulin completely for this child. And she's on 0 insulin now. Tablet dose has gone up. What happened to her blood sugar? That's in blue. Look at the blood sugar. This is when she was on insulin, no sulfonylurea. Blood sugar is 300, 400. As the insulin dose came down and stopped, and the sulfonylurea went up, sugars have become normal, completely normal. So she's responding to the tablet, but not to insulin. It's opposite of type 1 diabetes, okay? So this was fantastic. So we withdrew the child. So you may say, how long did she respond? Maybe 1 month she responded. No, she responded much longer, okay? So this is, and of course it made big news at that time when this was the first child discovered in India in neonatal diabetes, and we were the first center to set this up. So it made big news. This was Ivana Das. They've allowed me to use her name. They said, please go and distribute this information all over the country. The parents are so happy. That's her mother and that's her uncle. And that's a child who came in 2004 when she was 3 months old, okay? In 2023, December, she came back. She's 9 years old. She's now 9 years old. From the time she left her hospital 9 years ago, now it's 10 years. 10 years, she's not had a single shot of insulin. Not a single shot. She doesn't even do blood sugar testing because all her sugars are normal. We've got branches in Calcutta, so she follows up in our Calcutta branches every 3 to 4 months. Completely free of cost. She doesn't pay a single rupee to us, and she's growing up very well. Now, what benefit did Ivana get by genetic testing? If she had not done the genetic testing, by now, she would have taken, by 9 years, now I'm not adding the 10th year now, 1 more year has passed, 13,140 insulin injections she would have injected herself, 4 times a day. Calculate that into 365 days into 9 years, okay? 13,000. She would have spent 1,75,000 on the insulin. If you had given her the cheapest insulin in India, she would have spent 1,75,000, okay? Glucose monitoring, she would definitely have to do if she's injecting 4 times insulin. So she would have spent some more on that. And she would have spent 2.5 lakhs of rupees she would have spent in 9 years' time, just to be alive, okay, if she had taken her insulin. The monthly income of the father is 20,000, okay? So, thanks to genetic testing, all insulin stopped, not one shot, that means 13,000 injections stopped for her. Cost of the neonatal testing, we do it for 2,000 rupees. Abroad it's done for 1 lakh. 1 lakh to 1.5 lakhs is the cost. We do it for 2,000 rupees. But I'm happy to tell you, that not for a single child with neonatal diabetes have we collected 1 rupee. We've done it completely free of cost. Not only for Ivana Das, but for the whole country. Any child, anywhere in the country, just sends us a blood sample. It will be done free of cost. In 2 to 3 weeks' time, the result will be given. The child doesn't have to come also. Just the blood sample is enough. And from all medical colleges in the country, we get blood samples, right from AIMS Delhi to PGI Chandigarh to JIPMER to every medical college, every child who has type 1 neonatal suspected is sent to us. Now, cost of sulfonylurea, what did she spend on the sulfonylurea? For 9 years, she spent 6,000 rupees, okay, instead of that cost. So the total amount spent by the family for 9 years is 6,000 rupees. You can divide that by 9, you can divide it by 12, then you'll know some 100 rupees per month or something they're spending on the tablets, and saved all this money it saved. If genetic testing was not done, if she was not referred by her physician to us, I'm not even sure that she should have been alive, because sugars would not have been under control with insulin. She probably have died. At best, if she was alive, she would have 13,000 injections of insulin. Now, imagine my life, I want to find out every child with diabetes in India and convert them from tablet to insulin. That's my lifetime ambition. Of course, we've been doing a lot of studies on this, and just to show you the first series of studies, before sulfonylurea, you can see the HbA1c, how high it was, and after changing to tablets completely, look at the blood sugars while they are on insulin and after converting to tablets. Now, this is just a series of different genes, how they respond. You can see blood sugar 600, 700 coming down, year 1, year 2, year 4, completely normal. It's like almost a cure of their diabetes, okay, just by doing the small genetic. And here are other examples and other genes. So each gene, each variant, we know how they'll respond now. We have formed a directory of all genes now and how they will respond. Now, opposite condition of this is called congenital hypoglycemia. Child will go into low sugar, low sugar, low sugar, low sugar. The child will develop fits, okay? Initially, because glucose is not given, something is not given, but then it'll go on and on and on and on. And by the way, in neonatal diabetes, it's not just the diabetes that the child have. 50% of those children also have fits. They have neurological disorders. They don't grow up. They have mental retardation, because in the brain also the same receptor is there and same genetic defect is there. When you use this sulfonylurea tablet, even the brain symptoms go away. Everything goes away once you do detection. Same thing happens in neonatal hypoglycemia. The low sugar, they'll have fits, they can die if you don't treat them. In the earlier days, what they used to do was every child who had this congenital hyperinsulinism, they'll send them for surgery. Pancreas will be removed. The pancreas is producing too much insulin. So they'll cut off 90% of the pancreas. Imagine a newborn child, 90% of pancreas is also removed. After a few years, the child developed diabetes because insulin is gone. We have removed all the insulin. It was a terrible disease to treat, okay? So now we have the genetic defects. We have a full panel to detect all types of congenital hyperinsulinism. Once you detect this, there are simple drugs like sulfonylurea. It's called diazoxide. So from the very low sugar, here the opposite. There you saw the blood sugar coming down. Here the blood sugar is going up now, okay? From very low blood sugars to almost normal blood sugars. With different genes, the response is slightly different, but these are ones who respond to diazoxide. Here is another group of children who have been treated. So we set up the first monogenic diabetes registry in this country. It's completely set up by us and it's completely free of cost, as I said. And here are all the places from which we get the samples. That's our genetic lab in Siriseri, and for some of you who'd like to, we have an MOU with SRM, so some of you want to come and learn these techniques there, you're more than welcome to visit. This is a 6.5 acre campus where 40,000 square feet facility we built entirely by ourselves, and so you can come and, what happened? Can you remove that? Something got stuck. Okay. Right. So you may say, how many such children have you seen? Almost 2,278 referrals we've had now, of which neonatal diabetes close to 500. The CHI, the hypoglycemia, is also close to 400. MODI is 1,300. Genetic syndromes, 150. What is genetic syndrome? it may look like type 1 diabetes. Okay, they may need insulin, but it's not type 1. It's a genetic syndrome associated with type 1. Some of those children will develop liver failure and kidney failure, and they will die. It's worse than type 1. So if you know it, you may say, well, some of them you can treat, some of them you can't even treat. If you know, at least you can tell the parent, get one more child. This child is not going to live beyond 15 years of age. Child will die. Definitely will develop liver failure and die. There's Walcott-Rollison syndrome and many other types of diseases. So when you do the type 1 diabetes also, we just do off a screening to see whether they have anything else other than plain type 1 is fine. With insulin you can treat. So you can see that diabetes is not just one disease. So if some of you want to send samples to us, you can just scan this QR code, or you can take Dr. Radha Venkateshan's number and take a picture of this if you like, and just send us a sample. We won't charge you a single rupee. We'll do it completely free of cost for you. And these are my contact details. On every social media I'm active. And if some of you want to join my WhatsApp channel, you can join. You can just scan this, and if you scan the QR code, it'll just say join, and you just join and you can be on my WhatsApp channel. The advantage for this is that every day you'll get an update about something new in diabetes, which is happening, something new in related fields. I keep teaching people. So that's it. If there are any questions, I'd be happy to take. Thank you very much. Be allowed any questions? Or? Yes, sir. Or a mic? You can give it a mic. Yeah, you can ask anyway. Okay.
One is if you find some genes where you want to knock down some genes.
Okay. Okay. Fantastic. I'll give you my card and I'll take yours and we can. Okay. Okay.
I have published a lot of papers in
Okay. Okay. Yeah. Okay.
And one of the simplest thing I thought whether I should tell you while telling you or not, is that one of the ways is when you know that neonatal diabetes has occurred, maybe just to withdraw them from insulin if someone is very shape.
It's dangerous, sir, because if they happen to have there are other genes like the GADA gene and the insulin gene mutation. If they have those, they can develop ketosis. Then they behave like type 1, although it's not type 1. That other 50% may die. What I would like you to do is, if we have a point of care device. Now see, it takes 3 weeks. They have to send the blood sample. Then we have to do the genetic test. We have to do the bioinformatics. We have to do which thing. Then we have to send it to them. 2-3 weeks is lost. And during the time, we ask them to be on insulin because we don't know if you withdraw insulin, the child can die. If they have these two only, they can respond. So if you have a point of care test where you just put a drop of blood and tells you whether it's KCNJ or ABCC8, if it's able to say that straight away, then you can stop the insulin, and that's something which.
So one of the things which can be done nicely, that if we really believe in 1 or 2 or 3 genes which are really causing this, you can think of pancreatic specific promoters driven shRNAs in normal mice. So they will suppress the gene. Or if you want to overexpress something in a pancreas specific manner, you can do, and you can have a wonderful model of animal where you can do whatever you want to do for
Sounds wonderful. I'm not an expert in mice, but I will definitely get my team to interact with you. I'll take your card and I'll give you my card.
Thank you.
Thank you, sir.
Hello, sir. Nowadays, we see many people particularly in village side, they don't even know these types of diabetes. for them, diabetes means they have to take insulin. They don't know type 1 or type 2 or NDM, all those things. And without, at the age of 50, 60, without proper diagnosis, they simply take insulin. What will be your advice for them?
Above the age of 30 or 35, 99.5% will have type 2 diabetes. And in type 2 diabetes, less than 5 to 10% need insulin. They can be controlled with diet, exercise, walking, taking tablets, whatever is needed. So they very rarely need insulin. We don't expect the patient to know whether it's type 1 or type 2. At least if the doctors know whether it's type 1 or type 2, it's good enough. So if they have any doubt about the type, see, you don't have to do genetic testing for every villager who is 50 years old. Ask them to walk, diet, take medicine, reduce weight, they are done. You don't need genetic testing, you don't need any of that. In young people, in children, it's a different thing. If they are type 1 and they don't take insulin, they'll die. If it is not type 1 and they take insulin, it may not work. So in such things, they should go to an endocrinologist or a diabetologist who will be able to, a general physician may not know. So such children, they're not very common. See, we're talking about hundreds of cases in the whole country. They should go to a specialist who will then say, this needs this kind of thing. And if any additional tests are needed, there are centers like ours where it can be done. So it does not mean that the common man, in Tiruvallur or somewhere who's 45 years old has to go for genetic testing and do complicated things. All they have to do is to follow their lifestyle, sleep on time, do some exercise every day, cut down carbohydrate, take a little bit more protein. If they're overweight, reduce their weight, and some simple tablets for some time. If they are needed, they can take. So it's very simple to treat 80-90% of the diabetes. It's in the remaining 10% where people like us who have spent our lifetime on this have to apply our mind. Sorry, sir? Symptoms. The symptoms of diabetes in children are they start passing urine, excess urine. They'll be drinking water all the time. They'll be giving water, but they'll be still getting up and passing urine all the time. They lose weight. The type 1 diabetes, for example, 5 years, 6 years, they start becoming thin. And at that stage, if it is not diagnosed, the sugar can go very high and they can go into coma. And even when they go into coma, they may not think of diabetes. They'll think it's meningitis or encephalitis or something. They won't do a simple blood sugar. It takes 5 seconds to do a blood sugar, 20 rupees, 5 seconds. Any place. So we say that glucose should be the 5th vital sign. When you go to any hospital, they'll check your temperature, they'll check your pulse, they'll check the blood pressure, they'll see whether your breathing is okay. Add one blood sugar also into that. Cost 20 rupees, one prick just on the bedside, 20 rupees, high, low, you can find out. Okay, if it's in between, then you can send. But they won't come in coma with the blood sugar 120. It'll be 700 or 600 or it'll be 40, low. So you can very quickly in any clinic, any hospital, any rural area, you don't need a lab, even small glucometer is enough. It's life-saving. So we say child, adult, old person, anybody, when they go to any hospital, any clinic, just do one blood sugar. Just like taking temperature and pulse and all, check this also. This is the 5th vital sign. It's not expensive, it's available, it's so cheap. You must just think of it and make it a routine. Then you won't miss the diagnosis. So many children must be dying in villages, not yet diagnosed. That's a pity. So all our life we are trying to educate people to be aware about it. That is why we go to social media, because doctors cannot reach everyone. Social media people are watching on their phone. So then we educate people through social media.
Yes, thank you. Thank you for the excellent talk, sir. So my question is like, is diabetes, it's going to be only controlled or can we cure people from diabetes? And then second one is like, what is your thought about the stem cells and its role in diabetes?
Yeah. So in the case of type 2 diabetes, if you detect it early, when the beta cell function is still quite good, and if they've got some weight to lose and the duration is short, they can even reverse it and bring it to complete normal. It's still not a cure, because they put on weight again, it will come back. But still, it's as good as a cure. In type 1 diabetes can only be controlled. But by controlling, people are living 60, 70 years. So that is also good. Your stem cell comes for cure of type 1 diabetes, where you produce beta cells from stem cells. That's where technology and biotechnology comes in. You get stem cells, either adult stem cells, embryonic stem cells, convert them. They're pluripotent. So you convert them into beta cells. And those beta cells, you infuse them. And then for a year or two, they'll keep on producing insulin. Still in its infancy, but 2 or 3 have already been cured. In China, 1 case. In Boston, 2 cases have been cured. So it's beginning to occur. It will take another 5 to 10 years before it will come. But I'm sure that type 1 diabetes may be cured in my lifetime. I'm hoping. Neonatal diabetes, it's as good as cure. The child is just taking half a tablet and does not even do self-monitoring. With that itself, she's growing up, she's become normal. So there are many types of diabetes where as good as a cure you can get. For type 1 diabetes, this 4 times, 5 times insulin today has been completely replaced by an artificial pancreas which you've got. It's a pump, modern pump. The pump will find out your blood sugar. It will decide how much insulin to give. It has AI built in. So it will give the right dose of insulin. You will not get low sugar, you will not get high sugar. It's like an autopilot, the plane without a pilot or car without a driver. It's already happened. More than 1,000 children now we have who are on this pump. And they don't need to take 4 injections of insulin. Just fit the pump and it does the job. But we are not making it in India. So we should make it in India now. It's cost 6 lakhs of rupees because it's an imported one, monopoly. Why can't we do it 50,000 rupees in India? Copy the same technology and do it here, like the Chinese do. Why not we copy and do it here? And give our, every child with type 1 should be given this pump. Then they don't need to inject themselves 4 or 5 times a day.
Sir, it's a very exciting and scintillating lecture as always. I'm curious to know about the diabetes and deafness in one form of, it's very interesting. Can you please share your thought?
It's a mitochondrial diabetes, maternally inherited diabetes with deafness. So it's a mitochondrial defect, which the mother only will have. It's inherited through the mother. Father will never get the disease. And as you're sitting through taking the history, the mother will say, child's hearing is not very good. Immediately, what about your hearing? No, I also have a hearing problem. Finished. It's mid. So then we treat it. They usually need insulin, but we know, at least we make the diagnosis and we can give them some treatment for the deafness as well as treatment for the diabetes. At least we know what type of diabetes. We know the exact defect. It's in the mitochondrial gene where this particular mutation occurs. It also runs in families, but it's inherited through the mother, not through the father.
Thank you, sir. Thank you so much.
Okay.
Excuse me, sir.
No, one more. Okay. Should give ladies a chance. Okay. You want to use the mic? Yeah.
So the maternal diabetes, right? Does this affect the offspring?
Yeah.
The early and late diabetes, does this affect the offspring?
Yes. It affects
How does it affect the offspring?
Because the genetic defect, which is a mitochondrial gene defect, is passed on. You're talking about the deafness one you're talking about.
So the maternal diabetes, the mother gets.
The gestational diabetes.
Yes, sorry.
Gestational diabetes, we think is nothing but type 2 diabetes, which occurs during pregnancy. See, what happens is, you may be destined to get type 2 diabetes when you're 40. Okay. Now you're 30, but you're pregnant. When you're pregnant, you already got the gene. The gene is already there in your body. Normally, you would only got it when you're 40. But now, pregnancy produces severe insulin resistance because as the baby grows, all pregnant women, not those with GDM alone, pregnancy itself is an insulin resistant state. As the baby grows, the placental hormones and the lactogen and cortisol and all the hormones will start getting secreted because the baby is growing. Because that severe insulin resistance will set in every pregnant woman. Those who don't have a genetic defect, those who can produce enough insulin will not get GDM. They'll keep on producing more insulin, more insulin. If you already have the genetic defect, then as insulin resistance comes, the sugar will go up because the pancreas is not able to put out more insulin. The moment the delivery occurs, insulin resistance is gone. So the sugar will become normal. So they become normal. But they will develop diabetes because already the defect is there. Within 5 to 10 years, within 5 years, they'll develop diabetes. And that is why it's a great opportunity. Once you know GDM is there, you should follow up those mothers, see that they don't get diabetes, they don't put on weight. After the delivery, they'll put on weight. But they're not bothered about themselves, bothered about the child. So they don't stop exercising. Then they'll say, for 2 people you have to eat now. So they'll eat more, they put on weight. Okay? Mother-in-law will say, you just be here. Why you want to go for exercise and only be here? So then afterwards, she develops diabetes. So we are missing an opportunity to prevent diabetes. And if you prevent diabetes, the mother can prevent in the child also. So it's a wonderful opportunity to prevent diabetes.
Thank you, sir.
Thank you,
Thank
sir.
you. Thank you. Okay. Last
Excuse me, sir.
Sir, as I have seen, there are many branches throughout India you are having. Suppose there is one branch in Kanpur also. So do they do any counseling there or just sample collection only?
We don't have a, you're talking about us, we don't have a branch in Kanpur. We have in Calcutta, we have in Siliguri, we have in Bhubaneswar, we have. Entire South India, we have everywhere, including Puducherry. We don't have a branch in Kanpur.
Okay, so exactly
but we,
what,
maybe in the future, yeah.
okay, exactly what we do in the branch.
Yeah, so exactly, we can't do, see all this advanced research and all, we can't do in all our 50 branches.
Exactly.
So what we'll do is, but every branch is trained by a doctor whom I have trained. We follow the same protocols. So if a child comes there with neonatal, suspected neonatal, the blood will come to Chennai immediately.
Okay.
So same day it will come, by courier it will come. Any special test not done in any of those branches, it will directly come. We have the same electronic records.
Yes, sir.
Real time. So what is happening in Siliguri, sitting here, I know what is happening. The whole thing is one system only. Okay, but we can't set up 50 labs, 50 genetic labs and all. We can't set up. Setting up one is difficult. So we centralize the whole thing and do it here.
Thank you.
Thank you.
So much.
Thank you so much.
Thank you so much for the amazing talk, sir. So there's an announcement. There will be a special quiz that's conducted tomorrow, and there are 3 exciting prizes for the winners. So all of you pay attention to the speakers, all the lectures, so that you can win the special prize. And now we can break for refreshment sessions, which will be for a brief 15 minutes, and we'll be back here for the talks. The refreshments are served opposite to the hall.
I'd like to invite Dr. R. Satish to introduce the first speaker of today.
Welcome back, students. I'm happy to introduce Dr. Subir Majumdar. Sir is the Director General of Gujarat Biotechnology University. Before joining GBU, Dr. Majumdar served as a Director of National Institute of Animal Biotechnology in Hyderabad and was a Distinguished Professor at National Institute of Immunology in New Delhi. Dr. Majumdar is a Fellow of National Academy of Sciences, the Indian National Science Academy, and the Indian Academy of Sciences. So he has recognized by several awards, including L.C. Bose Fellowship and Professor U.S. Srivastava Memorial Lecture Award. These are very less from the more awards that he has got. He has kindly agreed to attend our conference in spite of his busy schedule as a Director General. Today he will deliver the talk on genetically engineered animals for the benefit of humans and animals. So you guys are having this animal biotechnology classes. So he wants to motivate the kids. I said like it will be very good. Our students will be excited to listen to your talk. So he agreed to the talk. So thank you very much, sir. So now I invite you to this.
So, good morning, Chairman Satish. Thanks for bringing me here. And it's difficult to see from here, but that's fine. I know why Deepak was asking. No, that's fine. Don't worry, I'm fine. I can manage. No, that's fine. Don't worry. So, how much is my time? 30 plus 10. 30 I'll finish. नहीं क्या है 30 मिनट बोलना है 30 मिनट का टोटल टाइम है
30 minutes is your presentation and then like 10 minutes for the question.
Okay, fine.
So there will be discussions.
Fine. So good morning. I'll be talking about genetically engineered. This slide also you can turn. Genetically engineered animals for the benefit of human and animals. And these are my colleagues with whom I work. I worked at National Institute of Immunology, National Institute of Animal Biotechnology, and now at Gujarat Biotechnology University. Today's talk will be more focused on students. So we all know that what is transgenic mice, because I think that BTech, MTech, PhD students are here. So basically, majorly, it was a technique derived for taking the human genes and inserting them into the animals so that when these genes go to the animals, you have a model in which human genes are expressed and you can really find out how the human genes work in a biological system. So we should understand that some of the many health problems affecting both human and animals are allergies, ulcers, arthritis, measles, birth defects, influenza, cancer, hypertension, tuberculosis, glaucoma, asthma, diabetes, several diseases, and many infectious diseases. They also commonly affect all of them. So we all know that we cannot experiment on human. That is why we choose human beings. Just now, Dr. Mohan gave a talk, and there are several models of diabetes in various animals, including lot of studies of genes in even zebrafish and C. elegans also, how the glucose homeostasis makes a life. So these are all done in animals. You cannot experiment with the human beings. Now, we all know that element models are essential for understanding underlying mechanism of many diseases. Basically, animals are used for pharmacokinetics, dose response, toxicity and efficacy studies. But now there are very, very advanced models of animals which are really suppress the bone marrow of mice and insert human bone marrow cells and kind of make a very nice humanized mice with dendritic cells, beta cells and T cells of human origin. There, for example, if you want to study cancer, then you can really take some cells of a cancerous person and make a tumor on this body. So then you can give any kind of vaccines and therapies, and body is actually humanized body because T C cells, B cells are that of human. Now, let us imagine that we want to have a transgenic animal, but you have to understand why we need a transgenic animal. So let us take a normal mice. If you try to infect it with poliovirus, you cannot, because they do not have a surface receptor which will accept the poliovirus and take inside the cell. Exactly like what you have seen in the case of the SARS-CoV-2. Do not talk. Students, don't talk. Do not use cell phones. If you want, you can go out. There is no problem. Right? Don't talk. So what happens if you see this is the receptor which will actually is required for receiving the virus and internalizing in a cell, in a human being cell. But in case of mice, this receptor is not available. So when people wanted to infect it with polio, it did not get infected. So what they have done, they have isolated the receptor gene of the human and then made a transgenic animal. So now mice is expressing human receptor on its surface. Now if you infect it with a polio, it will not only cause poliomyelitis, but it will express several disease-like symptoms like paralysis and other pathological changes. So that means a transgenic mice is to be made. It has to be custom made. You can take human genes, insert in mice, and it will express those human characters. Now you can have a human model of disease. That's very, very important for us. What I said was this. Now, what is utility of transgenic technology? So we can know genetic basis of human and animal disease and design testing and strategy for therapy. So most important knowledge has come from doing reverse genetics. Now there are a lot of advanced, gene sequencing knowledge you have, equipments you have. From there you can know about the genes, but you do not know their functions. So what you do basically, you do reverse genetics. You can insert that gene into an animal, and let us say the animal becomes bald. So you understand now that the gene was required for hair growth. Or animal develops diabetes, as sir was saying. So this gene is responsible for causing diabetes. It is impossible to evaluate progression upon treatment in lab animals. Therefore, what we do that we make a disease model in mice, rabbits, and genetic manipulation is very, very useful. So why I am saying, what do you mean by when a patient comes to hospital? Let me tell you in a different manner. When a patient comes to hospital, he already comes with a disease. थोड़ा पेन हुआ, paracetamol खा लिया, और थोड़ा पेन हुआ. After 6 months, 8 months, you go to doctor, they say you have a major tumor in your liver, for example. By that time, that tumor has occurred. But if you can find out a gene which causes the tumor, then you can make a mice model, and after birth, at the age of 45 days, it develops tumor. So you can study why and how tumor develops in a mice. Because guaranteed, this gene is going to cause a tumor at 45 days. So what happens then? You can make biomarkers of tumor development, and much ahead of your disease, you can find out that this is to be prevented. This person is going to have a liver tumor. That you can never do in human patients, because they come with an existing disease to the hospital. So understand that is another advantage. So for students, please, nobody is going to teach you in this way what I am trying to whatever I had difficulty in learning, I have put it on actually slides for you to understand it much easier way. So whenever we talk about a gene, it's present somewhere in chromosome this, and it's present if you uncoil in some portion, it is present. So what people call, there has to be a promoter which will drive the gene, and there has to be termination codon, so the gene's story finishes here. So he said there are 28,000 genes in human genome. That means here, how do you find out 28,000 genes? A promoter is the beginning, and termination codon is the termination of the gene. Then somewhere next there will be another gene and another gene. Now when you go to machine, it only reads promoter sequence, termination codon, gene 2650. Next, gene 2651. If you ask machine what is the function of it, doesn't know. It can only tell this is the gene, you find out a function. So you can basically cut this and insert it in an animal and find out what is the function. Is it causing problem in brain, liver, kidney, ureter? You do not know. You will come to know the function then. So basically, what is the promoter? Is the engine which is going to drive a gene. This pipe is what is most important. You call it exon. It is going to make mRNA and then going to, for example, make protein. This is the only important thing, but you need a promoter which will drive it. And you need a termination codon which shows finished, done. But actually meaningful thing is this pipe. So what these people are doing, they are carrying over this pipe for functionality. Now, whenever you want to express a gene, you need a promoter and you need termination codon. Remember, this promoter is very specific to a tissue. So your chromosome in your brain and your gut is same. But brain is not secreting trypsin. Gut is secreting trypsin. Because brain does not have promoter activated in the brain, which can actually ask it to produce a mRNA for trypsin. But gut has its promoter, which is activated in gut. That's why you make trypsin in gut. Glial fibrillary acidic protein present on the glia. That promoter is active only in the brain, not in your gut. So remember, our chromosomes are same in all cells of body, but tissue specific promoters get activated in a particular tissue and they drive a gene. So promoter is very important for driving a gene. So what we know, the machine gives a knowledge about promoter, termination codon, and lot of exons and lot of introns, but they don't know where this train is going. You and I have to do lot of transgenic work, generate animals or in vitro cell cultures and find out what is the function. If you want to know true function, you have to generate the whole animal. Now, so what we are doing, we are cutting the genes from a species or from other animal, and this is the native genome of a mice. We are going to paste it here. So this red color is pasted here, and now we see how the function of this chromosome or gene is changed. That is what is transgenesis. So remember, in fertilization, there is a male and female pronucleus after fertilization. Most of the transgenic animals are made using this premise. You have to insert gene of interest, that cut gene with a promoter into any one of these pronuclei, and when the cell divides, it's a single cell, so every cell will get that gene of interest, and the animal produced will be transgenic animal. So what they do, isko center mein click kariye. Boss, click this in the center. So this is what we routinely used to do at National Institute of Immunology. Now we do it in NIAB. This is a super ovulated several hundreds of eggs of mice. This is the pronucleus. Now the foreign gene, small pieces of the genes are being injected here. This will swell. As soon as it goes into this small ball, it will swell. It goes down, and then you go, the technical officer or technician will go up and take hundreds of eggs are here, suck another egg, insert gene, suck another egg, insert gene, and do like this. Several hundreds of eggs are injected. If you keep it for 2 days, 70, 80 of them will die. 20 will remain alive. You put it into the uterus of a pseudo-pregnant mother. She is not pregnant, but she is mated with a vasectomized male. She has a false feeling of pregnancy. So when you inject these eggs into that, some of them will attach, some of them will survive, and some of them born will be transgenic if God is with you and the gene is integrated while cell division in the embryo. But this is the technology which is used for transgenesis. Now, this is the equipment. So nowadays around 1.5 to 2 crore rupees worth of equipments are required. The other way of doing transgenesis is cloning. You have seen how Dolly was made. You have to remove the native nucleus, throw it away. You can culture cells and insert whatever genes you want to insert in them. And whichever cell is showing you integration of the gene, you can take that nucleus and put it in a enucleated egg. Iske center mein click karna boss. So what happens? This is a large animal, buffalo. So what we did, we published this paper. Sorry. Please click at the center. Not working? Leave it. So what we do, center ka nucleus we will remove and throw. And there are a lot of cells which are transgenic cells lying here of the same species. We can take nucleus of that cell and insert. So now you have a transgenic nucleus. So the whole egg cytoplasm supports the development of that nucleus. That's how cloning is done. Dolly. So the mother cells were taken from here. That nucleus was inserted in a enucleated egg. Another dolly was made because it remains same. But in between you can do transgenesis. So what we did we inserted green fluorescence protein gene. Anything now, if it is born or made, should be green. So when we have taken the cultures of oviductal cells of buffalo, transfected several cells. Out of that, for these two cells become green, we took the nucleus and injected in an enucleated egg. So if now the embryo is made, embryo should be green. Ideally, buffalo's embryo is not green. So you can see out of these two, one is green. This is the inner cell mass which will make the baby. Same way, out of three, one is green, indicating that transgenesis can be done in this way also. But your understanding, you need lot of technical expertise, 2 years of training, equipment for making such animals. First animal made in India was that of HBVX gene. So there is a liver cancer, hepatitis related liver cancer, all of you know. So there is a gene, hepatitis B virus X gene. So if you cut that gene and make a transgenic animal, that was done at NII, and first mice of the country was a mice with that gene overexpression causing excess size of liver and liver cancer as compared to a control mice. And that was published in PNAS with ICGEV. So while knowledge about gene sequences are increasing by leaps and bounds, we have really very less transgenic technological methodologies which have come into picture. So genetic engineering is a significant change from past method of developing animal types which were based on crossing and selecting from existing usually related organisms. So long ago what people used to do, uska बैल अच्छा है, इसकी गाय अच्छी है, उसकी तबीयत खराब नहीं होती, तो दोनों का mating करके बच्चे पैदा करो. Why? Because those will be good, because they were infected, they were healthy. From there we have now come to know the genes which are responsible for good health. So now you can do genetic testing and take out good animals and breed. So remember, the largest amount of milk producer of the world is India. But per animal milk production of Indian cows is 10 times less than the Western cows. If they produce between 15 to 35 liters per day, our cow ranges from 1.5 to 18 liters. So if per cow milk production can be increased, we will become very rich country. Just if you do that. That can be done on the basis of genetic knowledge. Now, I will not go into detail, but lot of people have made environmental pick. So there are overexpression of phytates which can utilize phosphorus efficiently, and lot of gases are made by the animals which are dangerous to environment. So now this transgenic pig is very environmental friendly. We all know that large salmon has been made, and in Canada now it is given, and now people can buy transgenic salmon which is much bigger in size and you can eat it. So genetic engineering technologies, and the first product was approved for the human beings was etrine, and anticoagulant derived from the milk of genetically engineered goats used for the treatment of the blood clots. So a lot of people have blood clots, heart attack, pulmonary embolism. This is a life-saving drug made by transgenesis in goat. Large animal transgenesis, this is called as a horn, and now people have used this same embryo technology, and what they have done, they have changed the gene, so now cows are made without any horn. During transportation or fight, lot of cow injuries were there. So one of the biggest visible thing is that all these cows' lines do not have horn. So they have manipulated the gene. So from this simple technology, you can really use this embryo way to insert the gene of your choice and can design the transgenic animal of your choice. Now, there are several, I will just show that there are several kinds of animals made here. I will come here to read it. For increased growth, pig, salmon. For larger ratio of the fatty acids, pig. For the reduction of the environmental impact through phosphorus and nitrogen release reduction, environmentally friendly pigs by transgenesis. Avian influenza. So Roslin Institute has made a chicken which will never have influenza. So it will not transmit it to human being, what we call as zoonotic diseases, because chick is resistant. There is a gene inserted, whenever virus comes, tries to replicate, it cuts the mRNA of the virus. So virus cannot be formed. Then there is a mastitis resistant animal and so many others. So don't worry about that. I'll also not go into detail as time is less. And this is very recent one. ANP328 gene was removed to make a better chicken model which can be resistant to the, again, bird flu, that is influenza virus. This is a far better model than what was published around 13 or 14 years ago by Roslin Institute. And what it does, I will not, again, if you want, I can give you reference, that in normal chicken, if you give the avian influenza, they will get infected, and you will find the chicken will be, all are infected. All chickens are infected and they will eventually die. If you take the transgenic animal where transgene is present and infect it with avian influenza, they will not transmit it to other generation and the chicken will remain alive. So it will not go to human also. Same way, these pigs are made, what is called as African swine fever. See, it's a very bad disease. Now you can make pigs which are resistant to African swine fever. I will not go again into details. All of you know, and if you do not know, understand, that mice doesn't have this receptor, which is a mice ACE2 receptor. So mice can never have SARS-CoV-2 related disease called as COVID. So you can never experiment and find a drug or vaccine. So what people did, they have taken humans ACE2 receptor gene and inserted it into mice. How? With a promoter which is specific to lung epithelial cell gene. So engine they have taken from lung epithelial cell gene promoter, and the pipe they added is human ACE2 gene. I have shown you the diagram. And they make these mice during COVID time. So what happens now? These mice cannot accept SARS-CoV-2, but these mice with a human variant of receptor expressed can receive and can develop disease of COVID-19. Now you can use this as a model to test your vaccine, to test your plant-based therapeutics, to test any kind of drugs you want to test, that how to get rid of this disease. So what we have learned until now is that for making humanized animal models for human diseases, or for animal models for animal diseases, or for better environment, better growth, better meat, more egg, you have to use this procedure of embryogenesis. This embryogenesis procedure is very cumbersome. As I said, you need around 2 crore rupees of investment for lab, need 2 years of training to the technicians to manipulate embryo. You know how embryo looks? Just thin mucus, like our saliva. Out of that, small bubbles are made. That is egg. So whenever you try to inject, it sleeps. You cannot inject. Very difficult procedure. It takes a lot of time to learn. You have to kill hundreds of animals by superovulating them with hormones, so that you have hundreds of eggs, because out of that 1 or 2 will be transgenic. Rest of them will not be transgenic. This procedure is a little cruel also. So it was very difficult to see what is the alternative. So can there be an alternative procedure for transgenesis without killing so many mice and without using assisted reproductive techniques? So if you think, we started thinking. I'll now teach you innovation. Not only research, but your B.Tech, M.Tech, right? And PhD. So you have to think innovation. What is an innovation mind? So you have to have a big mice colony, hundreds of mice, supervision, killing of mice, technicians, a lot of big 2 crore rupees of equipment. Everybody cannot afford in India. If in your department, if you want to do it, you can't do it. So we started thinking, can we help? Can we generate something else? So what we understood, that while thinking, it came to our mind that during cell division, there is a crossing over. And when there is a huge cell division occurring, people at the time of embryogenesis, when the cell division was occurring, people threw some genes of human. Maybe in mice embryo or maybe wherever. Those genes got incorporated because there was a lot of this crossing over happening. House change, like in a city, all of a sudden somebody said, all of you to exchange houses. So that's what happens during cell division. During that time, somebody's TV can go into other man's TV house, right? That kind of things can happen. That's crossing over. So during that time, they threw the gene. So what we started thinking, that anywhere where there is a cell division, you can think of creating a situation like transgenesis, what is done in the embryo. Now, I'll explain you this phenomenon. If you put certain drops of genes on my skin and expect that my skin will become transgenic, it's not possible. Why it is not possible? Suppose there is a program of Madam Hema Malini, and you find that police commissioner has come here. There's a lot of police bandobast, a lot of bodyguards have been sent by Dharmender to protect her. And those kind of environment is there. She just walks in, it's an advertised, planned program. And she starts dancing here for her solo dance. And I get very excited and I say, I also want to dance with her. So I try to jump on this stage and try to plan to dance with her and just come and jump on the stage. Police will come, put a gun on me, catch me, put it in a cab. They are just standing here, take me to jail. You cannot. It's an organized function. It has its own way of going. You cannot interfere. Then I get very frustrated in the evening. Somehow my friends releases me from with well, and then I go to a disco to ward off my frustration. There I see people are drinking and dancing. I jump there also. And I find nobody stops me. I'm integrated nicely and allowed to jump and dance with them. Nobody cares. So I am very easily integrated in a place where everything was haywire. Cell was dividing. They were crazy. They are dividing like hell. See, I have seen personally sea urchin egg dividing. You put the egg and sperm and you do a lot of lab work and keep on watching like this, what is happening on the screen. Cell divides like cell divides like that, madness. I can't imagine. I have seen, I am very lucky. So I am easily integrated because there are a lot of cell division were occurring. So we thought, other than embryo, which is the other place where a lot of cell division are occurring. In embryo, cell grows, it then makes body, nose, eyes. So, you can integrate your cell. Where else this kind of cell division is occurring in the body, and we could find out that embryogenesis is one premise and spermatogenesis is another premise. But in embryogenesis, only once the ovulation occurs in a month. Human beings produce every day 200 million sperm, every day. Can you imagine what a disco is going on in the testes? Every germ cell is dancing. See, 200 million sperm in a day. What amount of mitosis and meiosis must be going on in the testes? So when ovulation occurs once in a month, but spermatogenesis is a continuous process. So we thought in embryogenesis, they exploited cell division faster rate. Can we exploit in testes? So we inserted, so instead of females killing hundreds of animals, we thought we will take one male who will remain alive. We will try to insert a gene of interest in the testis. We do not need superovulated eggs, and same male can keep on mating with several females and make several founder transgenic animals. So we did it actually that we have inserted genes in testis and then we have given electric current. So when you give current, the germ says, like a fish, opens mouth. And as soon as it opens mouth, since the fluid surrounding it is having my dotted pieces of foreign gene, it gets integrated. And since disco is going on there, it tries to get integrated into the genome, like crossing over. And it worked for us. And we thought that when we give GFP into one of the testes and electroporate, and no gene insertion but electroporate other testes, under UV light, green fluorescence protein is made by the testes where we electroporated genes, not in the other one. Can you see very easily? This is the machine you need, and this technique is actually germ cell will open its mouth, and if I have circulated lot of fluid with foreign gene, it will get inserted, and we publish this paper in Nature Methods. This procedure was very good in our hand and very cozy, but this procedure was very dirty in others' hand when others tried to do who do not have expertise to do a lot of surgical procedures in the reproductive organs. Very often male became infertile because of cutting the wrong nerve. So now then we started working, and now we have discovered a procedure where you don't have to cut. Now you can directly hold the animal, inject the gene and give electroporation to both testes from outside. That will help in making large animals. When we talk about the mice, you can really have hundreds of eggs of mice by giving superovulation. Taking 12, 13, 14 mice. Each mice superovulates 30 eggs. Because वैसे ही mice को 12 बच्चे होते हैं. गाय को एक बच्चा होता है. Mice gives birth in 21 days. Cow gives in 10.5 months. Large animal transgenesis by superovulation is next to impossible, very difficult. You can make only few. But this testicular technology, if it works, you can electroporate a bull or a male dog or a male goat, right? And you can create a lot of transgenic animals. So it's a very good thing for making large animal transgenesis. And we using this technique, now we have to prove that this technique works as good as the embryo. So there is a gentleman called Samit Chattopadhyay, and he works with the SMAR1. And we knew we had this construct because we published a lot of papers with them. And we do make a lot of transgenic animals. I am not, with Deepak, we have paper with ACTRAC, Cancer Research Institute in Mumbai. And SMAR1 was the construct of Samit. So we took Samit's construct and make the transgenic mice using testicular root. It gave the same result which we previously published, that is spleen gets enlarged. SMAR1 is a gene which has role in immune related tuberculosis, all these things. Then we took a promoter. I said you INGEN. I'll give you a very good example. INGEN is tissue specific. Did I say that? Brain specific, liver specific. So we isolated a promoter which is skin surface keratin layer specific. So that K14 promoter we isolated from one of my students' own blood. His name was there. Now under that we tagged green fluorescence protein. If we generate transgenic animal with that, only skin surface should be green. So actually when we produce such animal by testicular transgenesis, this is the piece of skin, only the skin surface is green. Rest of the cells of the skin are not green because promoter is driven for the uppermost layer of the skin. So to make it close up, you see this layer only has the green. Other cells do not have greenness.
Last 1 minute, sir.
1 minute. Okay. 30 minutes go there. So like that we made. So now I will not go into detail, but I'll say using the same you can generate shRNA, what I was telling to Dr. Mohan, that you can insert shRNA and you can inhibit the gene which he is saying is important for diabetes and can find out whether it causes diabetes to the animal or not. Then you will have a wonderful model. Like that, we created a model for infertility, and we found that when you generate this infertile model, spermatogenesis is actually severely declined and sperm count is severely declined. That means transgenesis, you can make several human models not only by overexpression of genes like green fluorescence protein, but by shutting off genes, by stopping the expression of the genes in the cell also. So this is a very wonderful technique, and we do help. So future work we think we can do where we can take the human genes and insert into the spermatogonial cells, and you can have a, if there are major issues of human being where they say तुम्हारा बच्चा नहीं हो सकता है, उसको कैंसर हो जाएगा. If God wishes, you can use this testicular transgenesis in human also to delete that gene. And once you delete that gene and make a boy or a girl, for rest of the life, that family can produce babies without that chronic disease. Now, maybe, I said maybe, it's not sure. Now, I'll take 2 minutes more and show them very important thing. There are a lot of human disease where you need very costly therapeutic proteins. And those therapeutic proteins, this is the Guillain-Barré syndrome, this is osteoarthritis. Here you need human IgG, here you need bone morphogenic protein too. Their cost is extremely high. Now, they are 15,000 rupees a vial. So 7-8 lakhs for a set of treatment. Poor people cannot afford. So now, why? Because you make big industries, factories for culturing the bacteria or cells, and huge infrastructure, electricity, 3-shift labors, license, GLP, GMP facility. I have my own alarm. You tried to threaten me once. So animal bioreactor based, if you make therapeutic proteins, instead of the industry, if you can produce it in the milk, you need nothing. You need some 200-300 rupees feed to this animal, and then you can have a promoter of the udder gland, engine of the udder gland, tag human gene, and make a transgenic animal. In the milk of that transgenic animal, promoter will work in the udder gland. It doesn't see वहाँ पे pipe है, या गधा है, या घोड़ा है, या बाल्टी है. It carries. It will express that human protein into the milk, and then you can have it very cheap. So your cow or a buffalo is your industry, your labor, your electricity. So the cost will drastically reduce. So we did that. I will not go into details of that, but we isolated buffalo beta-casein promoter, and then we have made transgenic animals. Don't go into all these things. I'll only show that this, with this what we have generated, now we have started injecting udder glands, because if you make genetically modified animals, GMO has a lot of legal issues. So instead of making a udder gland whole animal, we now inject genes in the udder gland directly. You can see here directly we can inject genes in the udder gland. It will go inside, transfect the udder gland. Another thing, why it will transfer udder gland? During pregnancy, just at the time of birth, udder gland starts growing very fast. अगर आपने देखा हो, it grows in animals, it grows very fast to give milk to the baby. Again, cell division is disco is occurring. At that time, if you insert, you can make a transgenic green fluorescence expressing gland. And actually, we have used this to produce in the milk of mice and also in rabbit human gamma interferon. So buy human gamma interferon kit from market, test the normal mice milk, nothing is there. Test the transgenic mice milk, you are producing human gamma interferon. So this cost can be reduced to drastically low level. I will not go into more very high level molecular biology based therapeutic antibody production in the milk. We can do that. I will not go into detail. Some of you may know, read if you do not know what is nanobody. It is the most modern way of treating diseases. And we can generate nanobody in the milk, the cost will be cheap. I will not show you the molecular biology part of it. I don't have time. But what we mean to say that very costly therapeutic proteins can be produced at very low cost if you use this kind of technology. So instead of 14,000, you can make it in 100 rupees or even cheaper. So the cost from 100x can become 2x for this kind of technology. And we have to be smart, competitive, work for the benefit of the masses. And recently we have made an indigo chip, which is the first chip of all cows of India. So you have the whole genome on that chip. Now you can find any gene's function, any disease resistance from that chip, and it has become a very good thing from the animal biotechnology. For students, I will take 30 seconds. Try to be very bold. Don't run away from situations. Try to face, you see, no reason, ball is going somewhere else and he's afraid. Jump, jump, jump and take the opportunities like this. Take catches like me. Right? So what will happen that on one hand, we should keep refining perfect targeted gene integration, but we should go to masses and try to teach people that getting more from less. That means use animals, and from a cow, you get treatment for 1,000 human beings per day. So for a less expense, we must get more for helping the humankind, for helping poor people. And very importantly, can you read, everyone? If you can't read, I will tell you something. That believe in biotechnology, BTech, MTech. So biology is very, very important. Remember, gravitation cannot be held responsible for people falling in love. Right? Who discovered gravitation? Think, read again. Gravitation cannot be held responsible for people falling in love. How much on earth can you explain in terms of chemistry and physics? How much on this earth can you express in terms of chemistry and physics? So important a biological phenomenon as first love. You know what is biology? I'll teach you. It's not chemistry. It is not time. It is not physics. How? Put your hand on a stove for a minute and it seems like an hour. Sit with that special girl for an hour and it seems like a minute. That's the Einstein theory of relativity. So biology is different. Biology is not physics or chemistry. Biology is biology. So please keep believing in biotechnology and keep your dreams high and many. And God will have really going to help you, at least one of them in flow very high. Thank you.
Thank you. Thank you, sir. I wish I give you more time to play test match instead of continuity match. Yeah, thank you. So maybe one or two questions from the student side. So
ask us.
Hi, sir. Really interesting talk. I'm just curious that the transgenic animals that you make for high yield, how resistant are they to environmental fluctuations?
Which animal?
I mean, let's say the buffalo that you are engineering, how resistant would they be to the patterns of climatic change? Because the pattern of the diseases might also change with that.
No, nothing will happen. See, nowadays CRISPR-Cas9 has come. You can very targeted manner insert your gene in a place which has nothing to do with immunology. You have to do only that thing. For your question, you have to target the gene in a manner it doesn't go and sits on MSc class 1 or MSc class 2 expressing genes. It will not bother. And now there are a lot of targets people have made where you can fix that gene without bothering anything. Or you can be very smart. You can go to beta-casein producing locus and insert your beta-casein promoter driven human protein there, replacing buffalo beta casein. Going to do nothing. It is instead of buffalo beta casein, it will produce human beta casein.
One more question from the student.
Thank you for your wonderful talk, sir. My question is, we are trying to understand function of a specific gene by introducing into an organism. Is it always necessary that the effect we observe on the organism will be the same as we will observe it in the human, or are there any exceptions to this?
Yeah, up to large extent, because liver, kidney, brain, sex, reproductive organs, hormones are more or less same in mice and human. If not, there are certain diseases like early Alzheimer's, Parkinson's, or behavioral disorders. These are present in primates. So people have started using monkey as model for that. I understand you cannot have a psychological disorder in a mice, but you can do that having a transgenic monkey or Alzheimer's or Parkinson's. But majority of the disease, answer is yes.
Thank you, sir.
No. Excellent talk, Dr.
Yeah,
Mazumdar.
thank you very much. Thank
I'd like to pass a comment. I don't know if it has inspired the students, but for faculty, definitely your talk has inspired. You have mixed music and comedy and animation and everything, and you made a lovely presentation. Thank you so much.
Thank you, thank you. My whole way is that they have spent so much of money to me to give me air ticket and keep me in a hotel. So I must repay back, you know. So I think I have done. If you stopped me for tonight, I would have sung song for you also.
Now I requested Dr. Ilangovan to come on stage and felicitate Dr. Majumdar. Now I request Dr. Arunjit.
Not this one, the slide you are seeing. I thought you were talking about the plant. Okay, not this one. Sorry, I thought तुम क्या बोले तुम्हारा प्लान उसको दे दिया मुझे लगा नहीं sorry I know they made a mistake नंबर करके रख दिया यार एक दो
As we continue with the talk, may I kindly request everyone to maintain silence and refrain from any disruption. So for the next session, I would like to invite Dr. Aruljyoti Nagarajan to introduce the next speaker.
Our next speaker is Professor Deepak Modi. Is a distinguished scientist from currently scientist working at the National Institute of Research in Reproductive and Child Health, Mumbai. His research primarily focuses on reproductive biology, with a particular emphasis on understanding the molecular and cellular mechanisms that regulate fertility and reproductive health. His work has contributed significantly to understanding of role of stem cells, extracellular vesicles, and epigenetic regulators in reproductive process, particularly in female reproductive health. He has been honored as a Fellow of National Academy of Science, India, for his substantial contribution to the advancement of reproductive health research. Professor Modi continues to lead impactful studies that aim to bridge fundamental science and clinical applications in reproductive medicine. We are looking for a colorful talk. Thank you.
Very good afternoon to everyone, and thank you so much, Satish, for inviting me. And thank you, Dr. Devi, for this wonderful opportunity to listen to Dr. Mohan and, of course, Dr. Subi. Speaking in the same session is indeed an honor for me. I come from Mumbai. My institute is ICMR Institute working for reproductive health, and child health more recently is added into our mandate. And in my lab, my lab primarily focuses on understanding how the embryos implant, how the early placenta is formed, how things go wrong there to cause infertility and pregnancy-related complications. In addition, we do a lot of translational research. We have got into making organ on chips. You would have heard about it. We are making placenta on chip to make pregnancies safe. We are also developing tests to detect common infections associated with preterm births. What I'm going to talk to you today is about some of our work on a Y chromosome microdeletion testing, which we have developed in our lab, and also AI-based tool. And the work has been largely done by my postdoc, Stacy, was supported by PhD students Sanjukta and Shadan, and the collaboration is with Professor Abhishek Sengupta and Priyanka Narad from Amit University, and Priyanka is now at ICMR, and I thank all of them. I stand before you speaking about all the hard work that they have done. So this is a physical map of the human genome. You have all seen the karyotype, and all of the chromosomes are in pairs. In females, the autosomes are obviously in pairs with all of us, but in females, a sex chromosome are also in pair. You have two X chromosomes. However, in the males, the sex chromosomes are discordant. They don't have a pair, and you have a little Y chromosome. And let me tell you, this Y chromosome has largely been ignored. Half of the people sitting in this room have this chromosome, but men usually are ignored, and definitely I'll tell you Y chromosome is for sure ignored. You don't have to believe my statement. I'll show it for sure. This is GWAS, Genome-Wide Association Study. Sir spoke in the morning, where you know a lot of diseases are associated with chromosomes, and every chromosome in the human genome is associated with every other disease. But where is the Y chromosome here? Do you even see it? There is this little Y chromosome sitting there with almost no disease associated to it, as if it's one of the most useless chromosomes in the world. And you have heard the story of lately that Y chromosome is going to disappear and all the males are not going to. I'm sure females are all happy and smiling that this species will all vanish from here. But trust me, we are not there to go. I'll tell you why. So for the longest time, it was believed that the only job of Y chromosome is to make the males. Developmentally, the embryos have bipotential gonads, and they have to either form the testis or the ovary. The Y chromosome gives the SRY gene, the testis is made, and the business of the Y chromosome is all done, and the rest of the chromosome is useless. Having said that, it was in 1997, the first paper was published by Professor D.C. Page, which showed that that's not true. On the long arm of the Y chromosome, which has no other homologues anywhere, on any other chromosome, there are genes. These genes don't just exist, they're not pseudogenes, but they're functional genes, and they're expressed in the testis. And he discovered in total 12 genes. And this was a game-turning paper, which said that the Y chromosome is not probably as useless as we were thinking. It is having some genes, and if it is making some genes, it should have some functions. We did not understand the functions of many of these genes, but it's very interesting that last year, the complete sequence of the human Y chromosome was published, telomere to telomere, and whatever numbers of genes that we knew, 41 additional genes were identified, and all these 41 genes were coding genes, means they were proteins. So from 12, now we have the number to 41 plus another 28. So there are a good numbers of genes on the non-recombining region of the Y chromosome, which is now transcriptionally and translationally active, and it should have some purpose. So eventually, the genes were getting mapped onto the Y chromosome, and we already knew they were coming from the long arm. The short arm contains the SRY gene and some of the other genes. These are pseudo-autosomal regions which has X homologues, but the rest of the thing which was thought to be very, very useless, now we know that there are about several genes. This is a data which is a little older. This number has improved further. There are 28 protein coding genes. Some of these are transcribed but not translated, and there are 188 pseudogenes. And you can see that there are 3 regions in which these genes are distributed: AZFA, AZFB, and AZFC. And we were looking at these functions of these genes. When I started my lab, we were wondering what are these genes doing, and we showed that obviously they're expressed in the testis because we thought the Y chromosome should have a role in spermatogenesis and they should be expressed in the testis. And we did show that one of the gene RBMI, which is only present in the males, not present in the females, its expression is found right from spermatogonia to the elongated sperm. And then we looked at other genes like RBMY and DAS in fertile and infertile male. A lot of studies we did, even essentially to show that most of the genes which are there on the Y chromosome, the AZF, A, B, and C loci, almost all of them are expressed in the testes. That means they should have a role in spermatogenesis. And indeed, if we take these genes and do bioinformatics analysis to kind of predict their functions, we could predict the functions in a number of processes like regulation of translation, chromatin organization, regulation of gene expression, and a good fraction of them had known role in spermatogenesis because by that time many knockouts and transgenics have already been developed. So far so good. Now, we knew from literature that there are deletions of these Y chromosomes. The reason why everybody is believing that the Y chromosome is going to disappear is because there are human males who have regions of these Y chromosomes completely gone missing. And these genes are called as AZF deletions or microdeletions. You cannot detect them by a karyotyping. You need to do PCR to detect these deletions. And there are three distinct kinds of deletions reported in humans: AZF-A deletion, AZF-B deletion, AZF-C deletion. They can be in multiple combinations, and smaller deletions called as the GR deletions. And these deletions were almost always found to be associated with male infertility. These are males who do not undergo spermatogenesis. That means they're completely azoospermic, or they have low sperm count. They are oligozoospermic. So the question we asked in our lab was that what is the prevalence of these Y chromosome microdeletions in the Indian infertile males? Do Indian infertile males have these Y chromosome microdeletions? Is there a genetic cause of male infertility? Was the question we started asking with. So my student Sanjukta collected data from about 403 men who were either azoospermic, oligozoospermic, no sperm in semen, low sperm count, or low sperm count and poor motility. And out of all the men that we had screened by PCRs using 6 different markers, I'll tell you why the 6 markers were important. And we found that the frequency of Y chromosome microdeletion in the Indian infertile men was about 6.4% in men who had azoospermia, 5.8% in men who had oligozoospermia, and 3.2% in the men who had asthenozoospermia, and overall frequency was around 5%. Then we asked that, okay, about 5% of men who have infertility have Y chromosome microdeletions. Are there deletions beyond the locus which we were looking at, which would cause infertility in them? And then we focused on this locus called as the the AZF-C locus. Now, the C locus is very interesting because it is multi-copy genes. There are genes in several copies, and because this Y chromosome does not have a partner to pair in meiosis, it undergoes self-recombination, and while doing such self-recombination, it makes mistakes and removes some of these genes. So we asked whether these genes or these smaller deletions are responsible for male infertility in the Indian population, and we could, and there are about 3 different types of smaller deletions. Don't get bogged down by too much of it. They're called as GR deletions. They are called as B1, B2 deletions, or B2, B3 deletions. Don't get too bogged down. It's just that they're classified based on where the breakpoints are. And what we could show was that in the Indian context, the GR deletions were more strongly associated with male infertility. The chance of infertility was 4 times higher. We did find these deletions in the normal fertile males, in the normozoospermic males, but in the infertile males, its prevalence was way higher. And if you look at even the so-called normozoospermic males as compared to the non-deleted counterparts, the men who had GR deletions had lower sperm concentrations and lower motility, telling us that indeed these deletions are a cause of infertility in the Indian population. Then we figured out further and we thought, okay, can we narrow down? So this number was also additionally 2% was added to the whole business. We thought, okay, are there any gene copy numbers? Because I told you the multi-copy genes there. So we looked at gene copy number variations and we found that several genes were deleted. Copies of several genes were deleted more frequently in the Indian infertile males as compared to the control counterparts. And this is finally what we got. The yellow spaces means the gene is present, and the red bars mean the gene is absent. Each row is one gene of that locus, and each column is one gene of that locus, and each row is one individual. The block here that you see here is the map of oligospermic men. The block you see here is map of azoospermic men, and here is normospermic men. So there were several normospermic men who did have deletions, but there was one locus in whom we only found 2 normospermic men which had the deletion. Rest of them did not have any deletion, whereas these deletions were very, very common in the infertile male. So we now call this as a minimal critical essential locus for spermatogenesis. Now, what does that mean? That means that the Y is not going to disappear. Don't worry, you will be existing. So what is the proposed reason why it is believed that Y chromosome is going to disappear is because a Y chromosome was evolved out of taking genes from a lot of autosomes and made by itself. Now, over the time, it acquired mutations, polymorphisms, and it drove more genes there and became smaller, and then it started removing genes which it did not want. Obviously, when you set up a house, you get in a lot of things, but slowly when you organize yourself, you remove things what you don't need. So that is what is Y chromosome doing, and at some point people believe that in doing this business, it's going to disappear, it's no more going to need. But we think that after a point, so the present day Y is somewhat like this, we believe that deletions will keep occurring in the Y chromosome, but after this point, when it has reached a minimal critical point, infertility will start coming. And therefore, these men will not be fertile, and therefore they'll not pass on to generation. And a point will come, it will plateau down, and then the minimal critical Y chromosome required for human survival would be left. So that is what the theory is. Only time will tell who is right. Anyway, so this is all about fundamental research and all this. I mean, how does it matter clinically? I come from Indian Council of Medical Research, and when I present this to our SAC, they say, Dr. Modi, all is great and fine, but what is it for the patients? How does it change patients' lives? So we thought a lot. Obviously, when my brain is very fundamental research oriented. And when they told me this, I said, oh, look at this, such fantastic discoveries I'm making, and here they're more worried of curing men. Then I thought, okay, but I have to do it because it's a pressure on us, otherwise my promotions and my salaries won't come. So I said, chalo, kuch to karte hain. And we started digging back on our own data. So we thought, okay, let's do something which is clinically more relevant. And how can we change lives of these men? So then when we were looking at the Y chromosome microdeletion data, by that time Stacy had joined my lab as a postdoc, and we were looking at the Y chromosome microdeletion data in the world, and we found a very interesting pattern. Generally, most of the populations across the world reported a frequency of Y chromosome microdeletions to about 8 to 10%. 9 to 10% was the frequency of deletion that was reported. From India, it was 6.5%, almost half. And we were wondering, why should this frequency be so low? Why is the frequency of Y chromosome microdeletions in the Indian population so low? And then again, the genetic brains woke up and said, oh, there should be some haplogroups, some haplotypes. I was talking to geneticists like what Mohan sir taught, that, okay, maybe there are other susceptibility locus. We were digging and digging more and more genes, more and more genes. Nothing, trust me, was making sense. And my student Sanchukta was getting more and more frustrated because the PhD was getting more and more delayed. Every experiment she would finish and say, nothing is making sense here. ये तो कुछ लग नहीं रहा. Like, चल क्या रहा है ये? And I used to blame her कि तू ही कुछ गलत कर रही होगी. You must be doing something shit. So therefore you are not able to do it. You do it well. And probably almost for 2 years, we had a very bitter fight every alternate week. She would get me negative results. I would blow my top off and say that, hell with you. But then one day she got something very, very interesting observation. She said, within India, there are again 2 patterns. One is some states are reporting a frequency of 5%, and some states are reporting a frequency of 8%, and Gujarat was way away, 24%. I'm like, ye kya ho raha hai? this is interesting. And then again, the genetic brains woke up. She said, sir, abhi aapka genetics band kar do. Nothing of this is all bullshit. There is something more interesting here. Let me tell you. I told you that we use 6 PCR markers to detect these Y chromosome microdeletions. These markers are defined by the European Andrology Association. And if a Farang skin tells us that, okay, this works, we believe it works, and we just blindly start following it for our population. What she told me was that all those labs who are reporting around 4 to 5% or even lower have almost always used the 6 markers. Those who have reported above 7% have used more than 6 markers. This now rang bells to us that perhaps we are underdiagnosing the Y chromosome microdeletions in the Indian population. And then Stacy sat back and did a lot of calculations, and she realized that if you were using only the EA markers, the frequency was 5.9%. The frequency was 3.8% when we used additional markers. That means now is the time to define that what are the sets of markers we as Indians should use to appropriately diagnose. You can imagine 48% of cases, half of the cases were missed, underdiagnosed because of a wrong test that we were using. So you need the right kind of test and therefore the right kind of markers for detection of these Y chromosome microdeletions. So Stacey took up this challenge and said, okay, I'm going to sit back and figure out what are the deletions that are common in Indian population and which are the markers which we should use to diagnose them. So she did a lot of systematic review. She picked up about 45 papers from India. We collected data from our collaborators, Dr. Tangraj, with the largest contribution. Dr. Rima Dada gave us data. A lot of people in the country gave us data, and we statistically analyzed the data, and finally she narrowed down to a 16 marker panel. She said 6 will never work for us. You need 10 additional markers to appropriately diagnose our patient. And then she developed a multiplex PCR. It sounds very trivial here, but getting this multiplex PCR took us about 2 years because we wanted to optimize it. Now remember, we were wanting it not just to show that these 16 markers are enough, but we wanted the 16 markers for a test which people like Anderson could eventually use it to diagnose patients, because patients don't come to me for diagnosis. They'll go to a genetic lab. So it had to be a lab optimized test. And a lot of back and forth happened. And finally, we developed the 16 marker test where you can see that these markers are AZF-A markers are together, B markers together, C markers together, the GR and the positive control. So you can now simply count the numbers of bands and figure out whether there is a deletion or there is no deletion. And then you can see here this patient has all the bands appropriately, but only one of the band is missing, that we interpret it as GR. This patient, there are no bands in the B and the B and the C locus, and there is one GR band missing, so it's a complex deletion of AZFB plus C plus GR. So it is a very complex. So now it became way easier rather than running all these 14 markers separately and doing 14 PCRs per patient, which is way expensive. We narrowed down to a 5-reaction multiplex PCR, which allows a very rapid and a fast diagnosis. And with this, we now predict that all the missing cases that we have will be diagnosed. Eventually, we did a lot of studies. So we started at technology readiness level 3. We took it to around 8 in the span of 2 years. And then we had to negotiate with ICMR. And this technology is transferred to LPS, and it's now available commercially. Anybody who wants to use it can buy it and start diagnosing for the patients. This test is 100% specific, more sensitive than the conventional markers. Most of the available kits available in the market today use only the 6 markers or other random markers which are not appropriate for Indian population. This is a test developed by an Indian for the Indians. So then, currently, this test is being validated by a number of centers across the country. The final data is available with us. We are in the process of analyzing it, but almost all the centers have told us that this test has worked very well in their hands. Now, the question is, okay, fine, you diagnose infertility. Okay, there is an infertile man, you did genetics, you identified the cause of infertility. How is it important? How is it mattering? So first and foremost, when an infertile man, especially with low sperm count, comes to IVF clinic, they are treated with a lot of medications. They're given antioxidant therapies, they're given vitamins, they're given antibiotics with the hope that they will improve their sperm count. We say that those who have deletions will never improve by these medications, and they should not be treated with any of these medications empirically. They can straight be posted for a IVF. What we could also show was that men who have these Y chromosome microdeletions, as compared to infertile men who do not have these Y chromosome microdeletions, remember the residual spermatogenesis in the testis occurring, we can remove some of the testicular tissue and actually get sperm good enough to do a ICSI. I'll talk about ICSI in the next slide. But what Stacy showed was that these men who have deletions are twice more likely to get a sperm. That means now this test is not just diagnostic, but prognostic, whether this man is going to have some sperm in this testis for his future generation, or the man can never have spermatogenesis. So it was now a predictor of spermatogenesis in the testis. Now let us say that you did a TISA, testicular sperm extraction, and you did a ICSI. You remove this sperm, and as I showed, you can inject one sperm, and this is being routinely done in most IVF clinics, and it is a very successful procedure. Millions of babies have been born using ICSI. But now when you do this ICSI and make embryos, if the father has a Y chromosome microdeletion, all the male babies that will be born out of such a father will always have this deletion. There is 100% father to son transmission, and this has to be told to the patients. Currently, that's not being done in the clinics because nobody does this testing. The testing comes negative most often, and therefore these are underdiagnosed. So they're going to perpetuate infertility in the next generation, and here's a pedigree to show that the father had it, he did ICSI, he had two sons, both of them had deletions, and his son also had a deletion. That means socially, we are increasing the number of infertile population simply by not testing it. If you are doing a ICSI, you can tell the couple that, okay, you have a deletion, your sons will have a deletion, they will be infertile, and their sons also will be infertile. Trust me, many couples say, okay, we don't want a baby, we are fine with it, or we'll like to have a female baby, but you cannot do sex selection in this country. So they say that, okay, we'll opt for a donor sperm. But many couples come back and say that, so what? If you're giving me a baby, you will give my son also a baby and his son also a baby. Don't deprive me of biological parenthood. Now, this is also socially important. Everybody deserves for biological parenthood. So we said that, okay, this test can also help to predict the outcomes of IVF. So to summarize, Y chromosome microdeletion testing is clinically essential because these men will never benefit from any forms of medical therapies. They will not improve spermatogenesis. They can directly do assisted reproduction, and it will have father to son transmission. So this information has to be told to the patient, and we have released a policy brief. It's available on our website and with ICMR, where you can use this as an information sheet to talk to the patient. Anyways, let's get back to prediction of ART, success of assisted reproduction. So conventionally, if you open a textbook and now look at Y chromosome microdeletions, they'll say men who have AZF-A deletion and AZF-B deletion never has spermatogenesis. Don't let them do ICSI. Men who have AZF-C deletion, good chance of spermatogenesis, do ICSI. I can tell you that this assumption is wrong, and what we have now data to show that these Y chromosome, which I showed you originally had so many genes which are expressed, we also show that these genes are expressed even in embryos. XY embryos. Now, we thought that these genes were only important for the testis. We found that these genes are expressed by early embryos. You can see from morula, from 4-cell stage, they started the expression. They're there in 8-cell, morula, and then again they switch off. So transiently during embryogenesis, these genes come up. And what we could show was that if IVF is done with a sperm from man who has Y chromosome microdeletion, although he has a good chance of getting a sperm, his chance of fertilization, clinical pregnancy, live birth is significantly lower, telling us that you can do You can upload the picture of the gel and you can just say predict. And as soon as you say predict, it will give you the outcome. Here is the outcome. You can say that, okay, this patient has either a C deletion. So now the clinician doesn't have to bother too much of time to interpret the report. Using this AI ML, the interpretation has become easier, and it also gives the report of what is the chance of this patient with a deletion to get a live birth, what is the chance of his to get a sperm, a live birth, or a clinical pregnancy. And this has been copyrighted and it's also licensed to APS. So those who buy our kit also get this live software for free for prediction. And therefore, we think that this is a very user-friendly software. The clinician doesn't have to apply too much of brains, doesn't have to consult too many papers and books. It's a very straightforward thing. So we are now going from just genetics to actionable genetics, that, okay, if there's a deletion, you can not just counsel and say, don't have a baby. If you want to have a baby, this is your real chance. Now you take a call whether you want it or you don't want it. So to summarize, Y chromosome has large numbers of genes with housekeeping functions. These Y chromosome microdeletions causes infertility in about 10% of cases. That's the number we finally have from India. The tests that must be used, I'm not saying use our test, but anybody who is into the lab should use a test which is validated for the Indian population. If we don't validate it, you saw Sir's talk also in the morning, the Indian genomes are very different. The Indian genetics is very different. So it has to be validated for the Indian population, and such patients should not be treated with hormonal therapies. They'll have a good likelihood of sperm retrieval, but a poor outcome of IVF, and therefore it's important to counsel the patient, talk to the patient, and our AI-based software perhaps will help the clinicians and the patients in more effective clinical decision-making. Do I have 2 more minutes? Okay. So now I'm going to take you through of something that is food for thought. Up to now, I've told you that Y chromosome had key roles in sex determination, and it was useless. But now I told you that it has a role in spermatogenesis, important role in spermatogenesis, and therefore critical for male infertility, and therefore Y chromosome will not disappear. But evidence is now garnering to tell us that the Y chromosome has functions beyond fertility. It's not just a chromosome which is required for sex determination or fertility, but beyond fertility. And I'll quickly tell you why this is crucial. Because I told you that all the genes on the Y chromosome are expressed in the testis, but if you look at some of the other genes, they are expressed in every freaking tissue of the body. All of us who are XYs here, every tissue is making these genes, and all of the XX in this room don't even have these genes. You can imagine the difference in the transcription and the translational profile. Remember, I told you these genes have a role in chromatin remodeling. I told you they have a role in translation. That means the rate at which your genes are being made, other somatic genes are being made, the rate at which the proteins are being translated is going to be very different between males and females. And this has implications. Implications because I tell you that about 21% of Y chromosome genes are expressed in the liver, 34% in the kidneys, 34% in the stomach, and so on and so forth. So they're present in every tissue. And beyond infertility, it is now evident that gain or loss of these Y chromosome genes is observed in hepatocellular carcinoma. It appears that Y chromosome genes have a role in predisposition to cancer, suppressing or increasing the chance of cancer, and it is also has a role in testicular carcinoma. So beyond the reproductive tract, there is evidence to show that it has a role in hepatocellular carcinoma. Last year, there was a paper which showed that men who have cardiac disorders, especially cardiac fibrosis, they have mosaic loss of Y in the peripheral blood. So if you karyotype these men, there'll be some cells which will not have a Y chromosome. They're XOXYs. They've lost their Y chromosome over time. And a beautiful studies done in the mice have shown that if you have only the bone marrow hematopoietic stem cells, where there is a loss of Y chromosome, when these cells go and reside into the heart, stem cells always mobilize and correct our tissues, right? So the cells which do not have the Y chromosome, when they reside into the heart, these cells behave very differently and they induce cardiac fibrosis. So that means this Y chromosome not only has a role in testis, in the liver, but also has a role in the heart. And then these Y chromosome genes are expressed in the brain. And it is shown that men who have Y chromosome microdeletion, about 10% of them will develop neuropsychiatric disorder as they age. That means they have a function in the brain, and if you lose these genes, they'll be predisposed to neuropsychiatric disorder. And we have this very lovely case report. The father had a partial Y chromosome microdeletion. They did an IVF ICSI for this baby. The landed baby had a very large deletion developed because of the small, more errors were made in his spermatogenesis. The deletion became much larger, and the baby was born with mental retardation. So now it's not just important for the IVF clinicians to give babies to these men, but tell them that these babies might be predisposed to a lot of disorders. So to summarize, Y chromosome is not going to disappear because it's not just important for sex determination, it is also important for spermatogenesis and infertility, but it has perhaps a role way beyond what we have imagined, and there is a lot more to discover. I'll close here. I thank my collaborators, Dr. Indraja and late Dr. Kusum Zaveri, who gave us a lot of samples. I thank Dr. Jyotsna Gokral, who started this project with us, Dr. Rima Dada, Ashok Pal, Prafulla, Abhishek and Priyanka, Harsh, and the network of labs who are doing these assays for us. Thank you so much, and I'll be happy to take questions.
Thank you, Dr. Deepak Modi, for an excellent talk. Thanks for giving us hope that we are not going to extinct. So I'm happy. I have one curious question, like how many of the genes in that Y chromosome, you told around 40 of them. How many of them is transcriptionally controlled from the genes from the X chromosomes? Any idea?
Transcriptional control of these genes, we understand nothing. Okay. But many of these genes are controlled by genes on the autosomes. Okay. So we just discovered those genes. We don't even know how long the promoters are, what are the promoter elements. But for sure, there is evidence to say that they're controlled by autosomal genes.
Thank you. So now the session is open.
Oh, Deepak, nice talk. So I have two questions.
Sure.
One is, these deletions, you have seen more deletion in Indian population.
Yes.
Did you do the same thing in the other population? So they may not have looked into more number of deletion. That's why they don't report it.
Sorry, come again.
For example, you have taken more number of deletions. You have checked for more deletions, right?
From 6 you added more.
But the other countries, they may not have looked into the deletion. Do you have any comparison like that?
Yes, we are done, and they have all used more than 6 markers. And the prevalence is 10%. Okay. So 10% has been a universal number across. It was India, we were not getting 10%, and that started bothering us.
Any region-based, for example, African, Indian, there is a difference like that?
So by and large, if you look at the world map, I think one European nation and us, we had a low frequency, rest of it as 10 or higher.
Okay. No, not percentage-wise, number of deletions.
Number of deletions, so per man, it could be even one locus deleted or more than one locus deleted, doesn't matter. The number of genes which were used, number of PCR primers that were used were more than 6 in almost all these studies.
All these types. Okay, but also you didn't explain why Gujarat has this 24. Very high.
Everybody has been asking me this question. I don't know. We are
so I'm talking. Tests are more number of deletions or markers?
No. Now we are using this kit in the Gujarat population. Harsh has done this very systematically now, and the frequency is around 8.5 or 9, if I correctly remember. We are just waiting for this data. I think that one paper which came was probably messed up in a big way.
Thank you.
Can take one more question from the students. Boys, this is the time to stand up.
Yes, sir. I have one question. Like, do we can do something to come back with the extension of Y chromosome? Like, we have reserved tigers, and they are increasing in number. So, can we do something for Y chromosome also?
So, I don't think a Y chromosome will ever go extinction. It's the way evolution has occurred. To begin with the Y chromosome, to make the Y chromosome, a lot of genes were borrowed from the autosomes.
Okay.
Way before Y chromosome came into existence, everything was just in pairs. Now, what nature is perhaps doing is doing experiments that if I remove some genes, what is happening? So if you remove some genes and nothing happens, that means that that gene was useless for survival. Those which are now critical. So for example, in the minimal critical locus, I showed you that if you start deleting more from that number, you'll get infertility. So fertility will not continue, and therefore this genome, the faulty genomes will not continue. Okay,
Yes,
so
sir.
it's essentially pruning the Y chromosome to have the minimal locus that it needs for itself.
But currently, nothing is there in our hand.
I don't
Because
think we need to worry
IVF,
so much.
IVF is going to increase larger population, I think.
Yes, so that was the whole idea of developing this test and the tool that clinicians, number one, should be aware. Primarily, we tell them that do not offer IVF immediately to these patients, first counsel them. There are two groups of patients which are very easily seen. One group which tells that we don't want a baby, I'm suffering, I don't want my son to suffer. But other group says that no, I still want a baby, you can give babies to my sons, I don't care. So for both of them, but this counseling is very crucial.
Thank you, sir.
So is the scope of research for finding additional gene markers other than the 10 genes that have been founded to add into the gene panel specifically for the Indian population?
So we are currently thinking, we have been in the thinking phase for a long time. Technologies have limited us. We want to sequence the Indian Y chromosome because we don't even know structurally what the Indian Y chromosome looks like. Do we have more genes, less genes? So till that's done, I don't think we have scope. We'll have to buy by what's already there. So at present, my lab is analyzing those additional genes which are there, where what are they doing, what are their roles. But we are still going by the reference set to us by the Europeans and the Americans.
Thank you.
Thank you so much. Thank you for the thought-provoking talk. And more importantly, you gave all the men in this room a peace of mind. Hope. Yeah. And I would like to request Dr. Fiona from Anderson to present a memento to Dr. Modi.
Thank you for all our speakers for such an insightful session. Now I kindly request we'll break for a lunch and assemble back at the same venue by 1:45 p.m. sharply.